Hi, well, it's right at 8 a.m., so we'll go ahead and get the session started. My name is Lauren Hale. I'm a research microbiologist with the USDA in Parlier, California, and I have the honor of moderating the session this morning. So we've coordinated a session around the subject of evaluation and criteria for assessing biological inputs. There's been a huge increase in the number of biological products available in recent years, and there's a lot of promise behind a lot of these materials. However, there's sometimes some overpromise on what these materials could do, and maybe over guaranteeing of the kinds of outcomes you might see from these products. So as an organization, to address this, we wanted to coordinate a session around evaluation criteria for soil biologicals. So in our first talk, Patrick Brown will give us a broad overview of biologicals and their inputs and evaluation criteria growers can use if they're considering investing into these products. We'll also have some insights from industry. Jacob Parnell and Roger Bowman are going to share some background into what they do from an industry standpoint to develop these materials and test them in the field. And then we'll also conclude with a research perspective. Dr. Amisha Perret-Porterson is going to be sharing some background on her own research looking at organic amendments introduced into soils as a mean to modulate the native soil microbiota. So we start at 5, so it's up to you, Dr. Brown, if you want to wait till your alleged start or have a little more time for your talk and go ahead and launch into it.
I'm happy to start right away.
We'll go ahead and transition to our next speakers now. We have Drs. Jacob Purnell and Dr. Roger Bowman. They're joining us from Novozymes. If you want to go ahead and share your screens. Thank you again.
All right, are you able to see my screen?
Yes.
Okay, we've kind of divided this talk into 2 different sections. So Roger and I kind of work on different aspects of our kind of our pipeline of kind of these microbial inoculants. And so I'm going to kind of talk about some of the things we look for as we're trying to discover new inoculants. And then Roger's going to kind of take over and he's going to kind of explain how those are tested out in the field. So one of the interesting things that we see in the plant is that about 40% of the resources of the plant early on go out of the plant as exudates. And this, for us, this doesn't really make sense evolutionarily unless there's some really good benefit, because 40% is a pretty big number. So unless the plant is getting quite a bit of a benefit from putting all of these exudates out into the soil, then this would have, evolutionarily would have stopped a long time ago. And so we understand that there must be a lot of information, a lot of benefit that the plant can get from, for example, microbes. Now, a lot of the exudates are to try and solubilize different elements and nutrients, but a lot of it is to recruit a microbiome. And I've got here a figure from a paper that wasn't too long ago that really kind of shows how the microbiome shifts over time in the corn rhizosphere. And this is the figure itself, and one of our colleagues, Nathan Cude, has kind of put these shifts in terms of the growth stages of the corn plant. And so as you can see, you know, as we start to begin, we see this shift in the microbiome up until V3, and then as the plant matures, we see kind of this change in the dynamics of the microbiome, and we see that again, and these correspond to different aspects of the plant life, and this is over a lot of different soil types, several different varieties of corn. And so this is fairly a consistent model of what happens in the microbiome over the life of the plant. And so kind of one of the things that we're thinking is, well, maybe we can actually use some of this information. And so we did our own kind of very small study where we looked at just that first little bit of the rhizosphere microbiome in corn. And we saw an interesting pattern, because one of the things that we would like to see is rhizobium. Rhizobium are generally, well, they can be free-living nitrogen fixers, which would be great if we could get them to somehow work in coordination with corn. And this has been shown over and over that the corn plant will actually recruit rhizobium to the rhizosphere. And so this red line that I've got here is kind of a relative abundance of the rhizobia in the rhizosphere. So this is just to show, I don't have any numbers on here. This is just to show kind of the general pattern. But one of the things that we found is that we found that even before the rhizobium can kind of start to increase in abundance or be recruited, we saw something else that kind of looked a little bit like something that you would see in succession. Succession has been widely studied in macroecological settings. It's starting to be studied a lot more in kind of microbial ecology settings as well. And really, the idea is that you establish one species that kind of sets cues for another species to come in, and then you kind of can set the stage for what's going on as these communities are built. And so we kind of said, well, what if the same kind of approaches that you can take in, say, a forest succession can be taken in the rhizosphere succession of a corn plant? And one of the things that we found in forest succession is that you can actually kind of accelerate the succession by seeding the ecosystem with these pioneering species. So, right after a forest fire, if you go in and actually plant grasses or plant kind of shrubs, then that actually increases things, and so that you can get kind of old growth forest happening a lot sooner. So our question was, is there something in the rhizosphere community that we can add that can maybe shift some of our desired traits, the desired function of the microbial community to an earlier time, so that we're getting some kind of a benefit from the microbiome to the plant itself? So we really had two questions as we were doing this, and that was specifically for rhizobium. The first one is, can we accelerate the succession of this community to favor rhizobium? So can we put in something in there that's going to trigger an earlier recruitment of rhizobium in the rhizosphere? And then the second question really is, does that make a difference to the plant? So, if we do shift it, does that really benefit a grower later on in terms of their yield in their harvest? So, using some algorithms that we had developed, we identified some potential candidates for some of these pioneering colonizers. And we isolated a couple of these. And so what we did is we inoculated with these pioneering colonizers. We inoculated with just random microorganisms as well. And then we also had an untreated control. And here I'm presenting, this is the whole microbial community on this principal component plot. On the right, I'm showing kind of what the whole community looks like. And so at 5 days after sowing, we've just barely got a corn plant peeking up through the soil. And this is in potted soil in a farm soil. So this actually has a community in it. We're not using sterile soil in this case. And so what we've got here is these blue squares are showing the untreated control. So this is normally what the corn plant is looking like in that soil. And when we've got it with this treatment, with this pioneering colonizer, we see a different community in the rhizosphere. And as we progress, we start to see the untreated control actually starts to merge and starts to become more similar to the treated control. Now that's between 5 and 13 days, so that's a very short window. But one of the things that we looked at was, sure enough, as we add these pioneering colonizers, it looks like they are recruiting rhizobia. So these bar plots are showing it at 5 days and at 13 days, where, you know, at 5 days with this treated, we've got much higher reads of rhizobia in the microbial community. And then at 13 days, that untreated community is starting to catch up. So if I go back, we're starting to see that we are kind of shifting this over, so that by adding this colonizer, we're getting an increase in the rhizobium. So what does this mean, though? I mean, we're showing some evidence that we can do this, but really what we want to do is we want to be able to help benefit growers so that they're actually able to produce a little bit more. So these are in some greenhouse trials, and we're looking at chlorophyll content as a proxy for nitrogen content in the corn. And we saw an increase in chlorophyll content when we added these pioneering colonizers that recruited rhizobium. And even though we only have maybe an 8 to 10 day window where we're actually increased in the rhizobium in the rhizosphere, we do see that we have some benefit in chlorophyll content. We also did some microplot trials where we took this out into a small field trial, and we were able to see an increase in chlorophyll content in the shoots, and we were also able to see an increase in grains per cob of the corn plants as we harvested. So this is just one example of kind of a concept that we're starting to look into with regards to kind of these microbial inoculants. Another example that I'd like to present is with soybeans. And this is one where we started out similar, where we're looking at the microbiome of the soybean plant, the rhizosphere microbiome. And in this case, as we started to pull apart some of the interesting concepts and ideas within the microbiome, we saw that there was a functional group that seems to be important. And so this functional group is not tied to any taxonomy, so it's kind of widespread. So I've kind of got a fake little phylogenetic tree that I've built over here, and these yellow kind of represents maybe where this function is. So it's kind of spread throughout all the tree, but not everybody has it. But the ones that have it are the ones that are increasing within the rhizosphere. And so you can see that over here in our chi-square, where we actually look, and the p-value is pretty strong on this, where increase in abundance in the rhizosphere is tightly correlated with this function that these microbes have. So one of the things that we wanted to do then is say, well, if we just target this function and say, can we just take this function and then see if we have a greater proportion of these organisms that provide some benefit to the soybean plant, can we actually do that? And so we just randomly took strains from several different, because we wanted to get that taxonomic breadth, and so we didn't want to have everything within one group. So we took several of these strains and we inoculated them, and we, in this case, we did it with the Brady rhizobium as well. One of our products is a Brady rhizobium inoculant for soybeans, and so we wanted to see how it would work with and without this inoculant. And interestingly, we found that in most of the cases that there was some kind of a benefit to adding both of those strains together. So this combo is the Brady rhizobium as well as one of these functional microbes that we've added. So here I'm displaying 4 of these, but this was something that we were able to see over and over and over again. And as we decided, okay, well, let's look a little bit further into this, and we found one in particular where so this bottom picture here shows the pods from one soybean plant grown in a greenhouse. The set of pods on the left are untreated control, and the set of pods on the right are the bradyrhizobium and this functional together. And then this other figure that has 3 different sets of plants from our controlled environments show the untreated control, and then in the middle is our bradyrhizobium alone, and then on the right is the bradyrhizobium with this functional group. And so really kind of the things that we're seeing are that, for example, in corn, we can target a phylogenetic group to say rhizobium is important. How can we get rhizobium to the rhizosphere to benefit the corn plant? And then in the case of soybeans, for example, it's not necessarily a phylogenetic group, but it's a functional component where we can say, okay, we somehow, by adding this microorganism that has this functional component, we're able to get a higher frequency of organisms that can provide a benefit to the plant. And that's kind of where our discovery kind of side of things are going. And so once we've got these, then I hand them off to Roger. And Roger, are you on? Not sure if Roger is on right now. This is normally where he would take over.
I don't see him as a panelist.
Jacob, I can confirm Roger is not on as a panelist or an attendee.
All right. Well, I will go ahead and I will go through his slides, but with the caveat that I really don't know what I'm talking about. I mean,
Thank you.
I know what he goes through and I know everything he does, but as you know, if you want me to dive into the details, I will probably have to take any of the questions that you have, and I can have him respond back to you. It's a little bit of a busy time for him right now, as he's got several field trials that he's trying to get up and running. But so once we've got something that we think looks like a candidate, and I think this has been expressed quite a bit, is that there's a lot of difference between what we see in a lab, a greenhouse, growth chambers, and what actually happens out into the field. The last talk, you know, it expressed quite a bit of some of those problems of, you know, in the literature we see great examples of organisms that are doing phenomenal things in the greenhouse, but as soon as you try to get them into the field, all of a sudden you don't see that anymore. So there are a couple of things that we look for. The first one is our mode of action. So we need to understand how the organism operates, and this goes kind of along the lines of how are we going to design these field trials. For example, if we're looking for something that helps fix nitrogen, like I think it was Patrick before this talk that mentioned that if you're not seeing that stress in the field, then you're not going to see that phenotype or that recovery from that stress. And so you kind of need to know, what is it that we're looking for? Are we looking for, you know, is it an iron mode of action or a nitrogen mode of action, or is it a salt stress? You know, because if you put it in a field that doesn't have that, you're not going to see kind of that benefit from them. And so you kind of really need to identify those sites, those locations, and build your field trials around kind of that mode of action. And then also it helps to determine if there are any other factors. For example, you know, one of the ones that we were looking at with the corn study was this rhizobium. Does that, you know, 10-day make a difference? Does it make a difference if you're planting it early? does it make a difference when you are inoculating? Can you inoculate on the seed and plant it, or is it something that has to come at a certain time point during the growth of the plant? And then environment. So yeah, we kind of touched on this a little bit about how the growth chambers in greenhouse conditions are great, but they really need to be translated out into the field. And in the growth chambers and greenhouses, you can control so much more of the variability. You know, going back to the previous talk, which was fantastic, you know, talking about hot days. You can avoid hot days in a growth chamber or a greenhouse. You can have every day as a, you know, an ideal day. And then as we move over into the crop physiology, it's important to sometimes deliver it at the right time. For example, if it's a biocontrol, you know, you need to make sure that it's there when the pest is there. So it either needs to be something that can colonize and be there to prevent the pathogen, or you'll have to, you know, spray it at the right time in order to have maximum impact. So I'd like to drill in a little bit on the mode of action and try and compare biological to traditional chemistry. And so mode of action, in this case, a lot of the biologicals that we're looking at really are in concert with the plant. Whereas with chemistry, it's really a chemical reaction that you're trying to get. And so with the mode of action, it needs to be something that can survive on the plant. If it can't survive long enough to interact, you know, for example, as a biocontrol, if it's not something that's going to be on the leaf or on the root or wherever it is long enough to make a difference, then you're not going to be able to see that impact. And so as like the other big problem with biologicals is that they are very much more dependent on different environments. So on the environment. So, you know, for example, if we're going to go and we're going to spray something on the leaf, we need to make sure that our microorganism is UV tolerant, because the leaf is a very hostile environment for microbes. Another is it needs to be, you know, tolerant to desiccation. You need to make sure that you know, if it rains within a certain time after applying it, it doesn't wash everything off. And so a lot of the performance, a lot of the expression, a lot of what you're seeing is based on different environments that sometimes you don't have to worry about as much with traditional chemistry. Now there are some benefits in the mode of action for microbes in that you can be much more targeted. So, for example, we're working on a product that is a biocontrol that is very targeted for it's similar to BT, but it doesn't have kind of the downside of BT. So it doesn't have any negative interaction with you know, butterflies and moths, and no, it's with bees. I think it does kill the butterflies and moths. You want to kill those, right? Anyway, moving on to kind of the environment. As we start to look at the environment.
Dr. Purnell, just letting you know to wrap up quickly if you can, so there's question time.
Okay, I think I've just got a couple more slides, and some of these I've kind of already covered. For example, environment, you have to keep the microorganism alive in order to have it do its work, whereas with chemistry, they can usually handle a lot harsher environments. And the crop physiology, so a lot of times you could have with the biologicals, sometimes your microorganisms will interact differently with different varieties of for example, corn or soy. And so you kind of need to make sure that you've got the right varieties that are going to have that interaction, or at least they might have that interaction, but it won't be quite as potent as with others. And then finally, Roger put in this as kind of, this is what he would do to test kind of a mythical Uncle Ed's Wonder biological starter. So he would look for things like, does it increase fertility? Does it enhance root growth, promote plant vigor? Does it give higher yield? And then in order to test some of those, he would identify so mode of action, identify which nutrients are targeted, and then locate those fields where he could actually kind of test that to see, for example, if it's a nitrogen, then he would go out into nitrogen limited conditions so that he could really see the effect of the microbe. And then, I think we've covered most of these. I think he was just going to go into a little bit more detail as far as how each of these microbes or each of these inoculants are impacted by different environmental conditions, by different plant varieties, and kind of by different crops themselves. And with that, I will apologize to everyone for and ask you to excuse Roger. He's probably swamped with some field trials right now as we're speaking. So, but if there are any questions, please let me know.
Great, thank you so much. I think we are short on time for questions, so we'll go ahead and move on to the next speaker. But there are a few that have popped up in both the answered and open areas of the chat or of the Q&A. If you're able to answer those, that would be fantastic. And I think you did a great job covering Robert's section, so thank you so much, Jacob.
Okay, are we all ready to get started? It's 9:35, so we'll be starting the practical indicators of soil health session. And so the focus of the session is going to be discuss the biological, chemical, and physical indicators that are often used by both researchers and some other measurements that can be utilized in the field to evaluate how well the soil functions, and in saying how well it functions, how well it sustains the productivity of crops would be one very important metric. And so each speaker is going to talk about, how to understand and interpret different indicators in this context, can be confusing. And so hopefully they're going to be unpacking some of these questions that we might have. Our first speaker is going to focus on those biological indicators of soil health. And this is Lauren Hale, and she was the previous moderator in the last session. She's a soil scientist based out of the USDA ARS in Parlier. And Lauren, if you're all ready, I'll hand it over to you.
Thank you, May. Okay, so I'll go ahead and get going. Thank you all for joining today. I wanted to give a little bit of background onto.
Sorry, Lauren.
Yes.
Could you make sure your view is on the slide presenter view?
Yes. Okay. This one. Does that look better?
Yes.
Okay, thank you. Okay. So I wanted to give a little overview on soil health in general and sort of why we have this renewed interest in this topic. Certainly the idea of improving soil fertility, soil health is nothing new. Intercropping was a practice that's known to be used by indigenous peoples for many millennia. Over 2,000 years ago, Plato actually warned that soils were becoming skeletons with the fat and soft earth having wasted away. Early studies in the 1910s and the 1930s in the United States began to associate soil aggregation with fertility. And this is still one of the key metrics we use to associate a healthy soil. And then, of course, in the United States, we have our famous Dust Bowl. So we saw the ecological disaster that was derived from poor soil management. It contributed to the establishment of the USDA Soil Conservation Service in the 1930s, which is now the NRCS. And so we've known for millennia that it's important to value our soils. Currently, it's pretty widespread that we see soils as a source of a building material, as a filtration material, and a growth media. So why the sort of modern interest? And we see topics like regenerative agriculture, which consider soil health as the main component of those strategies. We see large organizations like the Soil Health Institute being established. We start hearing about soil health in popular documentaries and some government incentives. And I think a lot of this sort of rejuvenated interest comes in part in reaction to climate change scenarios. So we now see that soils, not only in poor management, can be a major contributor to greenhouse gas emissions, but also that they have the potential to help us with mitigation strategies. So this is a beast, and it won't be tackled with a single strategy, but multiple strategies can be used to mitigate some of these implications of climate change, and soils are certainly going to be a great sink of carbon if we use them responsibly. We also have a new capacity to really survey the soil biology. So before DNA sequencing became cheap, available, we didn't have a great understanding of just how complex and diverse soil microbiota were. But now it's fairly reasonable for scientific labs to take soil samples, extract DNA, and survey a large variety of the microbial organisms via taxonomic identification or via their traits. And we understand that soils are biodiversity hotspots, that they have some of the most complex biodiversity of any ecosystem we've ever studied. And so that's brought, I think, a lot of interest around the idea that maybe there's something to that. We also saw studies like Dr. Purnell presented, where a lot of plants' photosynthetically fixed carbon gets released through root exudates. And it's kind of hard to fathom why a plant would do that. We also know that microorganisms on plant root surfaces have lots of traits that contribute to plant development. And so maybe there's maybe one of the reasons plants release so many exudates is a recruitment tool. So we're sort of starting to get new tools to really examine these environments. And then we also have a new generation of consumers entering our market. So Generation Z is considered to be one of the most environmental conscious consumer bases, as well as one of the most human health conscious of the consumer bases. They're the largest growing network of consumers. They contribute, I think, an estimated 140 billion into the US dollars into the US economy. They're estimated to be 40% of global consumers. And in one survey looking at consumerism in Gen Z, they found that 92.8.
Lauren,
Yes?
I'm sorry. I think we're not seeing your slides advancing, or are you still on the?
Oh, I just haven't advanced.
Oh, I'm sorry. I apologize. There was just
That's okay. Thanks for keeping up. So Gen Z's 92.8% of Gen Z consumers, when polled, noted that environmental protection and human health qualities were important when they made their food purchasing decisions. So I think in responding to all of this new information and the new consumer base, soil health is not necessarily a trend for, the early 2000s. I think this is going to be something that is really pushed, and we can all kind of work together to see how we can respond to these consumer changes, to these climate changes, and the many challenges we're going to be facing. Okay, so investigating sort of what role microorganisms have in soil health. So in our soils, we have an entire food web, sort of the basis of which is the bacterial and fungal communities. However, it is actually very complex. You have soil nematodes, you have small macrofauna like rotifers that come through and graze on these organisms. So basically, as microbial cells take on nutrients, a lot of those nutrients are recycled back into the same system by these microbial graders. But this talk is going to focus mainly on the soil bacterial and fungal communities and their roles in soil health. I just wanted to bring up that this is actually a much more complex ecosystem. We just have a lot of research that focuses on the bacteria and fungi because we have better ways to survey them using their DNA. So bacteria and fungi can influence the way that soils retain water. They themselves can produce extracellular polysaccharides, which kind of helps glue soil aggregates together. Fungal hyphae are also heavily associated with the soil aggregate stability and overall soil structure. There are also key drivers of soil nutrient cycling. So many types of bacteria can fix atmospheric nitrogen. They can also solubilize phosphorus, making it plant available. They can sequester iron for plants, and also can be associated with trace nutrient uptake by plants. They also produce and interact with plant hormones. So many microbes that sit on the surface of plant roots can actually produce plant growth hormones like auxins, gibberellins, and cytokinins. So when you have these organisms on plant roots, you often see increased root elongation or increased formation of root hairs. So all of that increases the plant's access to different soil environments where it can maybe encounter different nutrients or water sort of pocketed within that soil. Some microbes also have the ability to interfere with plant stunting hormones. So basically they can degrade a precursor to the plant hormone ethylene. If that hormone accumulates in the rhizosphere, it elicits a stunting response in the plant. And so they've shown that microorganisms with the trait to degrade that precursor also reduce stunting responses of plants to different types of abiotic stress. And they can also reduce pathogen abundances. Some of this is just competitive displacement. Microbes are there taking up this, the niche that the pathogen may have normally fallen into. they are competitive for nutrients, but because they're competitive, many soil organisms actually produce antibiotic and antifungal compounds on their own. So it's considered that having more diverse and abundant microorganisms in a soil could help to reduce pathogen titers in those soils. They are the base of ecological food webs and contribute to the total soil biodiversity. So if we want to go in and look at, say, a different management practice, so a field that is cover crop versus one that is not, and we want to say, basically, is this cover cropping actually improving any biological metrics? There's several different metrics we can use, but I'm going to kind of give away the ending of my talk, which is that there isn't one single perfect metric that evaluates a quote unquote healthy soil biota. So there are many different metrics and they all sort of have caveats and limitations. Further, even a metric that works really well in one place is not likely to work well in a different climate, a different cropping system. There's a lot of seasonal and even sometimes short-term dynamics to metrics we use to survey the soil biota. So one metric surveyed at one time of the year may not work as well as that same metric surveyed at another point. And then of course, other factors like soil type, the depth at which you're sampling. There's a lot of things that are going to impact the ability of a metric to portray an outcome. So I'm going to go into some of the metrics that we do have and discuss some strategies. So we know that the soil biota are exceptionally important to a healthy and resilient farm production system. But we know that we don't have a great way to just come in and consistently track them and say, this is a healthy soil biota. So I'm going to present some different things we can do. So one is to evaluate your own site. So one of the single greatest challenges in coming up with metrics for healthy soil biota is coming up with something that's consistent across cropping systems and locations. However, if you're monitoring a management at your site, you can time this appropriately and monitor this consistently. And it may be that this indicator actually works really good if it addresses a problem that you're seeing in your own site. And another strategy is to take a more holistic approach. So rather than going in and addressing a soil biological property like the total abundance of microorganisms, you might instead look at a symptom. So a symptom of a degraded microbiome might be that soil has very poor infiltration. So this is a symptom of something much bigger that has a physical, chemical, and biological set of properties that's lending to say, a compacted soil. And so another approach can be to look at the symptoms and then use steps that are known to promote soil microbiota and then address if that symptom is being mediated when you're integrating those approaches. So in looking at some of the indicators that a grower might use in this first approach, I am going to suggest that farmers work as scientists. So farmers usually already do work as scientists. They know a ton about their farming system. So you're sort of your expert within this system. So I'm going to present some biological indicators that are available, but I would also really recommend a review paper from Noah Ferreira's group, where they publish this in soil biology and biochemistry. They go through a lot of these indicators in more detail and expand on what I'm going to touch on today. So one common indicator is to look at carbon mineralization rates. This is also sort of seen as the CO2 or carbon dioxide evolved from a soil. And so there are some kits available as well as testing centers that can provide these information for you. You might take a soil sample and send it into a lab, or you might, as our student here, Julio, is conducting this measurement in a field. But basically, this has an assumption that if you have more carbon dioxide being released from your soil, that is indicative of a more active and abundant microbial community. And by abundant, I just mean there's more cells, there's more living biomass there. And so one of the pros of this is that this is very commonly done, that it often links well to soil carbon and microbial biomass. However, the con of this assessment is that one, carbon dioxide emission is not always a great thing. And two, it's going to be very sensitive to seasonal changes. Soil moisture and nitrogen availability will have large impacts on respiration rates. And thus, if those things are inconsistent, you might just be measuring the inconsistency, say, for example, in soil moisture, but not actually have a good metric of your soil microbial biomass. So this is an example of a data set where we're using this in a table grape vineyard. And so basically we have cover crops in this vineyard. We have a Merced rye and a Phacelia cover crop. And then we have basically a weedy control. We're trying to keep this bare. But we wanted to make sure that after we mowed and left this cover crop in place, that we don't see large CO2 emissions in the alleyways as a response, because we're actually concerned of, we're more interested in the total greenhouse gas emissions here. So what we found is that across time, basically all of these soils are pretty dry. They're in the center alleyways of our table grape vineyard, and they're not receiving irrigation inputs. So we actually saw that after this cover was mowed, we didn't see increased CO2 from our cover crop treatments, whether that was a native Phacelia species or an introduced Merced rye species. So there's not a significant difference in these treatments. However, we do have higher microbial biomass, which we know from a different type of analysis I'll go into later. So what we can see here is, as we apply this cover, we do have stimulated microbial biomass, but because the soil is dry, we don't have increased greenhouse gas emission through this season. So that kind of brings me to our next indicator. This is just calculating measurements of total microbial biomass. So people do this in different ways, and there are commercial options for having these types of analyses done. Some way is to extract the phospholipid fatty acids from a soil sample. And so phospholipid fatty acids are associated with the cell membranes of bacteria and fungi and some other eukaryotes in the soil. But mainly through this analysis, we get an estimate of the total microbial biomass in a soil, and then we can even break that microbial biomass into some broad microbial group classifications. Another method is to fumigate the soil. So basically you extract the dissolved organic carbon from a soil, then you fumigate the same soil and do an extraction. And the difference in the dissolved organic carbon is an indicative of the microbial biomass that didn't extract in your non-fumigated soil, but once it was basically sterilized, these cells lysed, and that DOC from the microbial component became available in your extract. And another method is to inject a substrate like a glucose into the sample and then monitor to see if you have increased respiration. So these are all different metrics to get at the same concept, which is the total abundance of your microorganisms in your sample. So these tests assume that microbial biomass is indicative of a healthy soil biota, but that's not necessarily always the case. It can also be sensitive to seasonal variations and sample handling. Some pros are these are pretty widely used metrics, and they can be very informative of heavy degradation. So you will see most likely impacts of tillage and pretty destructive practices using these metrics. The major con of these types of analyses is that we're not really learning anything about the functions. So all of those beneficial traits I discussed, we're not tapping into and analyzing whether those traits have the capacity, whether the community has the capacity to perform those traits, or whether they are actually performing them. We just kind of know the size of the community. So here we looked at tomato plots that were treated with a compost and biochar amendment, just biochar, and those that were unamended. And we wanted to look at the yield of tomato even with different irrigation levels. So we have the lowest, a medium irrigation, or sorry, the high irrigation level, a medium irrigation, and the lowest irrigation level. So what we can see by looking at the microbial biomass is that we did actually dip down in our control soils, that basically are unamended soils that had deficit irrigation at about 50% of evapotranspiration returned, were significantly lower than those in the unamended soils that had full irrigation. But you don't have as heavy as a swing when you had compost available. And so this can be an indication of how these amendments are actually functioning to support tomato production, even with less water. So in this case, the microbial indicator just kind of helps us form a link between what this organic amendment is actually doing in our system. Another common indicator that you'll see is reports of fungal to bacterial ratios. So fungi are notoriously sensitive to things like tillage, and these ratios help to let us know the proportion of fungi to bacteria in our soil. This is usually achieved using that phospholipid fatty acid analysis I mentioned. However, there's also DNA-based techniques to look at these ratios and microscopy-based techniques to look at these ratios. So the assumption here is that a low fungal to bacterial ratio indicates a disturbed or unhealthy soil community. However, these can be sensitive to sampling location. There's a lot high variability within a plot. There's also seasonal variability in these metrics. So the pro of this technique is that we get some basic information on the composition of the soil microbiota, and we're also tracing an indicator of soil disturbance. But the cons is that we don't have great coverage of many trophic levels. So to really determine sort of a healthy soil biota, we'd be considering the whole picture, the biodiversity of organisms at many trophic levels. And this only gives us a picture of 2 of those groups. And another challenge is that in many field soils, I see this more in annuals, less with perennials, but oftentimes our fungal biomass is so low that we have a hard time picking it up with PLFA. And so for a lot of different types of crops where, tillage is basically a big part of the practice, it may be really challenging to even obtain this metric.
Hey, Lauren, could you
Yeah.
finish up in 2 minutes? Thanks.
Yeah, so I'm going to skip the next indicator. They're in the slides if you're interested. There are also metrics to look specifically at the mycorrhizae in soils and to look specifically at soil enzymatic activities. So these are commercially available, and I've listed some pros, cons, assumptions with each of them if you want to review the slides later. I do want to give a little time to touch on these taxonomic profiles. So sequencing technologies have emerged, and we're getting to a point where you can commercially obtain sequencing data by sending soil samples to different labs. This data tends to be pretty complex. It's tough to draw, strong conclusions from these data, but you do get a much bigger picture of, say, your overall soil biodiversity. So one taxonomic profile is to look specifically at markers associated with bacteria and archaea, and to use them to compare samples and assess indicators like biodiversity or community structure. And so there's a lot of specific ways that this data is analyzed and worked with. And so its main, I think, limitation right now is that the interpretation is hardly feasible outside of the field of individuals who specifically do this. However, this does give you a much broader comprehensive picture of who's there. It wasn't even a technology that was feasible 10 or so years ago. So these are efforts that I can see industry sort of potentially coming to meet demand if it's there, where you might have a qualified individual help review these data and help make interpretations. But again, generally with these types of data sets, we tend to just generate more hypotheses. We don't necessarily know for certain if some function is occurring just based on the DNA that's present in a sample. And so this is an example of looking at some of that community data. And like Amisha had shown, we use ordination analyses to help cluster our communities. And from those, we can see that, for example, compost and manure, microbial communities from compost and manure treated soils were very dissimilar from those that were unamended. And we can also see that in our unamended soils, when we applied deficit irrigation in a turf grass system, we saw much more dissimilarity between our two microbial communities than when we amended soils with biochar. So this is kind of just helping us to see big picture impacts of these amendments on our total microbial community structure. So addressing the symptoms, so this sort of symptoms holistic based approach. I'm going to give an example. So a vineyard, for example, might have low water productivity. So we're applying a lot of water, but we need more water to attain the same yield. Another example might be there's plant nutrient deficiencies and or high fertilization demand in a certain agronomic system. Another symptom might be there's high disease susceptibility. So each of these indicators are not solely based on biological processes, but biological processes are important for every single one of them. So in this study, we want to enhance.
Lauren, I'm sorry, we've got to wrap it up just so we're going to run into the next session.
Sorry about that. Okay, we can end there.
Sorry about that. So maybe there are several questions for you in the Q&A.
Yeah,
and we do have 2 minutes. I wasn't sure if does this session have the multiple choice questions, if we can have those put up? Oh, you don't. Okay. All right. Then in that case, sorry to cut you short, but I could just read to you one of the questions.
Sure.
What did you relate levels of organic carbon and biomass to soil depth? And also, what time of the year is best for sampling? Winter, spring, summer, or fall?
Yeah, so in the first question, with soil depth, you see a pretty sharp decline in soil carbon, and you see those trends with the biomass as well. So as you go through, as you go deeper into the soil profile, your soil biomass gets quite low, and it's actually really challenging to assess soil microbial properties from low depths. And then, what was the second part to the question?
Just what several people have asked, what's the best time of year to assess the soil health of a sample?
So often, and it's kind of a disappointing answer, but throughout the growing season. So things are shifting a lot throughout the growing season, and I usually select time points that are important for that crop. So I might look at nut maturity. I might look at, bud break. So I would usually choose a few time points throughout a growing season that are important in crop development. And I usually avoid measuring right at harvest, because at that point, I'm a lot less interested in what's going on with the soil microbes than I was for the period of time that fruit was really developing and ripening, or when the plants were first planted or seeded.
Okay, great. Well, thank you so much. And if you could type in your answers for some of those other questions in the Q&A section, that would be great. Thank you.
Will do. Thanks.
Okay, our next speaker is Sat Darshan Khalsa. He is a project scientist based in the plant science department at UC Davis. And Sat, welcome, and thank you for joining us.
All right, thank you for having me. I just want to acknowledge May and Karen Lowell for inviting me to give this talk. So if you're interested in soil organic matter and specific to orchards, you're in the right place. So I'm Satdarshan Khalsa. I'm a assistant professional researcher in plant sciences at UC Davis. And today I'm going to talk about soil organic matter specific to orchards and really trying to link those, link the soil organic matter within the orchard system and bringing in concepts of carbon and nitrogen cycling specifically. So, oops. So a brief presentation overview. So first I want to talk about a nitrogen study that we carried out starting way back in 2008. I want to talk about how orchards cycle nitrogen. And then I want to then extend within that same study, same orchard, we did a rate study. And I want to show how that rate study impacts the carbon balance within an orchard. And you'll see that it really focuses on soil organic matter, really being the key focal point for much of what I'm going to speak about today. Then, of course, within this realm of soil health, you know, there's a lot of different practices we can consider. I would say most of them are really aiming at augmenting, improving soil organic matter, and that really being a focal point for a lot of soil health measurements as well, sort of the big picture measurement. What are we trying to do on a larger scale? So I want to talk a little bit about organic matter additions within the context of orchards. I specifically will touch on compost mainly. It's been my focus in showing how applying compost and what's the end balance associated with where compost ends up after one growing season. And then lastly, I do want to touch a little bit on this idea of barriers to adoption. I know myself and most of the folks in this audience have this, you know, this need to then go out and work and talk with your constituents, your clientele, your communities about different types of practices, about soil health, getting people excited about it, really. And, you know, in doing so, we have to be cognizant of who we're talking about and what their potential concerns might be. So really growers being the main focal point for this piece. And then also kind of encapsulating a little bit more about like, well, what types of organic matter are available and what are some of the trade-offs associated with it. So I put this slide up. The slides that I'm going to present at the first part were a paper from last year. This is the big picture slide. Not to go into all the details here, but if we do focus here on the soil organic matter, the carbon and nitrogen, we see that it is intricately linked to a lot of different parts of an orchard, mainly through mineralization and mobilization cycles. And then of course, we see these normal processes of end cycling within the soil leading to denitrification losses, as well as leaching. And I'll touch a little bit on that today. But really, we want to focus on what is, you know, what kind of nitrogen is being taken up for tree, and when we're talking about efficiency. And unlike annual cropping systems, orchards have allocation to wood, as well as to the leaf canopy and to the crop, of course. But we also see like other crop residues, leaves cycle back into the system. And of course, you know, how does that also work when we're fertilizing a system like almonds, for example, that is highly productive and requires lots of nutrients and water to be so productive. And then, you know, linking it to the carbon cycle a little bit with greenhouse gas emissions, how does that then impact, you know, CO2 and the cycling of greenhouse gases related within an orchard? So first, I'm going to touch on this idea of orchard nitrogen cycling. So back in during my graduate school, we applied 15N within an orchard. And what we did in the first year is it was in July, so it was early July, late June, so it was a good time to get a decent amount of nitrogen applied in that orchard. And what we saw right away is on the x-axis here, we have the year, and on the y-axis, we have grams of 15N atom excess. So like, where are we able to trace the 15N within that orchard? So what we saw right away is that the largest N pool, when we went back right around harvest, was in the tree wood and the tree roots. We do know that almond trees and other orchard trees, vineyards included, store a lot of the nitrogen in the root system, and that's where it's being held before it's being necessarily translocated within the tissues for annual growth. After that, we then saw the second largest pool was the soil organic matter. So even though this particular orchard was very low in soil organic matter, it still has the ability to hold on to some of the nitrogen that was applied. And then lastly, the smallest pool, again, within this just brief cycle of applying the 15N, was in the, the crop and the leaf canopy. So in this pool over here. So what we saw, interestingly, after 2 years, around 2012 or so, there was a linear decline in stored 15N. So the 15N within the tree is starting to decrease over time. We start to see a cumulative growth in nitrogen allocated to the crop, and that's what this line is we're following here. Now with the crop, we have to consider that we're balancing all the nitrogen where it's going. So it's not the crop, just imagine it's a yield of almonds that's accumulating next to the orchard over time, and that curve we're seeing for the crop is a cumulative number. And then lastly, we see a rapid decline of the leaf canopy. So left nitrogen is building up in the leaf canopy. And likely what we're seeing in all these decrease, and as well as this net release of nitrogen from the soil organic matter here. And what we're seeing really is there's just a large allocation of nitrogen that's being stored in all these tissues into the crop. And that's what we'd expect to happen with almonds and other orchard crops over time. Now, keep in mind that this orchard continued to be managed with water and fertilizer. What we are doing, though, is we're tracing the 15N and where it moved within that orchard over a number of years. So what happened after 5 years? And this was really the most interesting finding of all. We see this continued decrease of stored 15N, and we see a leveling out really of the gains of the crop as well as the decline within the leaf canopy. But to me, the most fascinating thing of all is that the soil organic matter from 2012 increased back to the similar level right here as it did in 2010, 5 years prior. So to me, that was a really fascinating discovery. A lot of the work in 15N and cycling in orchards just showed a lot of what we showed here with the crop, but not really factoring in the soil organic matter. We didn't realize that the reality is orchards cycle nitrogen a lot like deciduous forests do. So we would expect through leaf litter fall, through root exudation and fine root turnover, that nitrogen actually begins to break down and then return back to soil organic matter over time. And that was a real key takeaway from this part of the study. So we then extended that to a 5-year rate study. We then looked at within that same orchard 2 nitrogen rates, one at 200 pounds of N per acre and one at 275 pounds per acre, keeping in mind that this orchard's soil organic matters were quite low at 0.35%. So after 5 years of managing these orchards and measuring different pools of nitrogen where we ended up, we found that the reduced rate increase, both rates increased soil organic matter, though slightly but significantly more for the intensive rate, so to 0.54%. The reduced rate, and then that was for soil organic matter and carbon particularly, but of course it has nitrogen, and we also measure that. For the reduced rate, we saw no change in the soil organic total nitrogen. But in the intensive rate, we actually saw a gain in total nitrogen of 165 pounds per acre. So on the right here, I have a table that's I converted from the paper and put into pounds of N per acre over 5-year units. And we can see the breakdown of the balance of nitrogen within this orchard. And looking at really this key take-home message here is that in the higher intensive rate of nitrogen use, we saw that there was higher net N immobilization. That is really the buildup of soil organic matter from higher intensity nitrogen inputs resulted in more buildup of total nitrogen within that soil. So when we applied more nitrogen, the orchard had more potential productivity. We potentially saw more of this cycling and turnover, and that ended up building the soil organic matter to a greater extent than when we used a lower nitrogen rate. Now in life, there are trade-offs, and it's important to recognize that these orchards that were also intensely managed had higher nitrogen losses, 185 pounds over 5 years compared to 95 pounds. How did that translate to end use efficiency, which is a really important parameter? Well, for the crop N recovery and total N recovery numbers for the reduced rate, we had very high percentages there, very highly productive, but also efficient systems. With the more intensive rate, we had a lower end use efficiency. So that's the real trade-off, and those were significantly different. But when we brought in this factor, what I called orchard N retention, which is really trying to factor in nitrogen within soil organic matter, that buildup of nitrogen in the intensive rate led to no more significant difference. So when we factored in soil organic matter per unit of nitrogen applied, there was no difference into how much nitrogen was either was taken up or retained in the soil. In other words, wasn't lost to the system. So how does this nitrogen end use translate to carbon cycling, and how is this soil organic matter really the focal point for all of this? We do know that when you intensively fertilize systems, you're going to increase greenhouse gas emissions. I'm showing that here in the same table from the same study. You get higher urea hydrolysis CO2 emissions, you get higher N2O emissions, and when you convert it into CO2, it's greater. But really, when you start factoring in the changes in soil organic matter, you're going to see that they're significantly greater for the intensive rate. And we showed that. And this is these units here from soil organic matter converted into CO2 equivalents. And what's interesting is when we're building soil organic matter, we were able in this specific orchard to offset all the other farm emissions, leading to a negative net global warming potential. And that was significantly lower for the intensive rate. So again, when we're in fertilizing with higher nitrogen, we actually stored more organic matter that offset more of the greenhouse gas emissions. And when we factored in, wood carbon sequestration, which was equal between both of the rates, we find that orchards in general are creating a net CO2 storage that's a higher, you can do more of it with intensive farming, that's likely driven by the tree wood, but also by some of the practices with how orchards are farmed, likely no-till and how trees grow and turn over, as I showed earlier. But this is not all, come with solutions. Anything in agronomy and soil science, we have trade-offs. So we can't necessarily focus on intensively farming all of our orchards to increase CO2 storage, because that comes with a trade-off of higher nitrogen losses to the environment. So that leads to the question, what if we were able to substitute some of the nitrogen inputs that are leading to losses with compost or other types of soil organic matter? So we have this ongoing compost trial in almonds. This is research that's ongoing. And it's focused in San Joaquin County. We have a few other sites around the Central Valley, but the longest site that I've been operating for research since 2015 and is in San Joaquin County. We have a grower control. So the grower is applying fertilizer like he would normally do based on his CCA recommendation. And the data I'm going to show you right now is based on the first 2 years of work, where we also applied compost at a rate of 4 tons per acre, which is actually kind of not a very high rate of compost application. The idea here is that we were going to try to continue to apply compost over time and to see what would happen if we did it on an annual basis. Now, we used 2 types of compost. We used a composted manure from a local dairy farm that had a C/N about 10 to 12. And then we used a green waste compost supplied by Recology, which had a higher CDN of around 13, and in 1 year is quite high, 18 CDN, 17.5. And in these compost treatments, they also received fertilizer. So the compost was in addition to the fertilizer that was being applied by the grower. Oops. So what measurements did I take for these first 2 years? What we wanted to measure was first release. How does nitrogen decompose from compost? So this is unique, of course, that the growers apply compost on the surface applied application. It's not tilled into the soil. So we wanted to really understand how growers are managing compost differently and how it decomposes. We measured the availability of organic N using resin cores. And what we were really interested also is how this changes soil total nitrogen and the soil organic matter. And then what I called recovery, which I'll show in the next slide, is the sum of inorganic N plus the change of soil total nitrogen. So how the nitrogen was changed from the additions of this compost. So this paper is currently under review, and the figure we're seeing here is a nitrogen balance. This 1-to-1 line is release equals recovery. So when we were able to measure releasing from the compost, we were able to recover. So that would be a 1-to-1 line. If you were below that line, the 1-to-2 release down here, that means that the recovery is 50% of the release, which is probably signifying some sort of inefficiencies in the system. And then, of course, we actually measured here, naturally wasn't expecting this, but what we saw is, well, what happens if you get greater than 1? That's where the recovery the nitrogen you were able to recover in the system was greater than the nitrogen that was released. So keep in mind that fertilizer was being applied. And if a compost in this case has a high C/N ratio, in this case 17.5, we were actually recovering some of the fertilizer that was being retained by the compost itself. So what we first wanted to understand was the total applied nitrogen as it was related to the release. So we applied about 130 to 170 pounds of nitrogen when you convert from 4 tons per acre. And about 75 to 85% of that was released. That was able to decompose within that growing season, which is pretty good. It was not affected by the C/N ratio did not affect nitrogen release. So we didn't see any effect where different composts broke down differently depending on, they all broke down approximately the same. But we did see differences, of course, is in net N mineralization, which is how much inorganic N was available. With that 10.5 compost, we were only at 3%. And to me, that signifies that there were definitely some inefficiencies there or some losses to the environment that we weren't able to capture. When we looked at those composts between those two lines of 12 to 13, then we started to see this 6 to 8% available. So the compost, the nitrogen is becoming available. Of what was applied, about 6 to 8% became available. And then lastly, that compost that was higher was only available less than 1%. It was very low and almost not available at all. What is important here, though, is this net change in total nitrogen in the soil organic matter. For the 10.5, it was only 32 pounds of, that was 20% was applied, fairly inefficient. For the 12 to 13, and this is the key take-home message, 75 to 100 pounds of what we applied, we could detect in the soil organic matter. That was more than 50% to almost 2/3 of what was applied was just, I was able to recover it just in the soil organic matter itself. So this is keeping in mind that compost application has to do with building soil organic matter, not necessarily trying to get that nitrogen out of it within that first year. So this is kind of an important take-home discussion point if you're speaking with growers or other folks in the future. For the higher C/N ratio, we were able to detect a lot of the compost in the soil organic matter, and we actually had greater than 100% of what was applied, likely because there was immobilizing the fertilizer nitrogen that was applied. So briefly, I want to touch on this idea of barriers to adoption. Back in 2017, Patrick Brown and I did a survey of all the almond growers in the state, asking about organic matter, how they use it, what are their concerns, priorities, so that we could then translate into research that would be meaningful for them. Bar none, it was food safety is the number one issue, number one concern. And we can see that here on the top panel and the lower panel. The top panel is users of compost who took the survey. The bottom panel is non-users. So people who I sent the survey to said, I've never used any of these amendments before, and then they also filled out the survey. So regardless if you use compost or not, you have a concern about food safety. I think we've demonstrated over the years that with proper compost and proper application methods and good breakdown and decomposition before harvests, that we can manage food safety in a way that we can still continue to use these practices. In almond specific, if we're looking at off-ground harvest or other types of technologies in the future, that's actually going to make it almost easier and more beneficial to try to use some of these practices because food safety is a top concern. Interestingly enough, the non-users, people who don't use compost, they're going to have this larger barrier of cost and logistics than a user would. And that makes sense. If you haven't used compost, you're going to probably think it costs more and you don't have the equipment and you don't know how to do it. So that's going to be more of a concern than the third concern that I outlined for them, which was nutrient availability. So people who use compost still have this concern about when are the nutrients going to become available? How is it going to impact my fertility program? And I hope in that slide previously, I showed that it really depends on the carbon to nitrogen ratio and how much you're applying and what you expect to get out of it. So lastly, I want to touch on quality and availability of compost. I want to say in this map here, we have manure and its rating of accessibility by almond growers across the Central Valley. And then this is green waste. I want to point out here, this in the San Joaquin Valley, manure and the availability of manure is a huge opportunity. It's accessible, it's abundant, but of course it's going to require partnerships with the dairy industry. And I'm involved in a few different projects now where we're taking a closer look at that. It makes a lot of sense, but there's a lot of different barriers that we're going to have to overcome. We all know that dairy farmers work really hard growing forage and producing milk and managing a herd, and asking them to make compost on top of all that that's good enough and certified enough for almond growing or other crop growing. That's just a whole new enterprise. So if we're going to work with the dairy industry, we have to recognize that we have to come with a lot of information and a lot of support in order to make that happen. Lastly, on the other hand, green waste compost is very valuable. Typically, it's certified. It tends to be consistent in its quality, but the challenge is it's less accessible because of its more centralized, composting facilities, and then in different parts of the valley, and that can increase transport costs. And the other part that I didn't mention here, of course, with manure, it's less available in the Sacramento Valley, so there's still going to be cost if we're going to be using manure in that region. Lastly, just want to briefly touch on technology. This is actually not a barrier for using compost. It's simple equipment. We're going to need more information and more partnerships, but in order to do this and do this effectively, we're not looking at really huge amounts of technology investments to do so. A lot of the equipment and ability to make compost is already there. Keeping in mind that this is, of course, a long-term investment in research and in farming. And like my colleague Amélie Godin says, it's a long-term investment just like your trees are. So quick summary, we did the nitrogen study and we wanted to show really that orchards cycle just like forests. The rates of nitrogen impact carbon balance, but there are trade-offs related with nitrogen. When we're adding organic matter within the first year, we're getting less than 10% nitrogen availability. However, about half to 2/3 of the N applied is showing up in soil organic matter. What does that mean for subsequent years? I hope to answer those questions for you all in the coming years. And for barriers to adoption, we still need to have this focus on nutrient availability. I don't think it's going to go away because we're all used to using fertilizers and how to manage those. And this creates a whole new different way of managing. And of course, we also want to consider the variability of different organic matter sources. And I think this is something that, with not only the almond or orchard industry, but other industries that are supplying need to be cognizant of. So here are the references for the papers that I've shown. And of course, I want to acknowledge lots of people that I work with, not enough time to name them all, but certainly the Almond Board, CDFA, USDA, FFAR, and UCNR for supporting this work and ongoing support. So thank you.
Great, thank you so much, Satdarshan. You have just a minute or so for a few questions, and then maybe if you could answer the rest in the Q&A during the next talk, that would be great. And just to remind everybody, there's no DPR or CEU Zoom polls for this session. So in regard to the compost study, someone wanted to know, was that 4 tons of compost that you applied, was that each year or only in the first year of the study?
So those studies, I did say 2015 and 2016. Though that work, it was like I repeated it. So in 2015, I applied 4 tons and then measured what happened after a year. In 2016, it was like a true replication. I went to fresh ground, applied the compost, and then did the same thing. So to answer the question, when we're looking at those numbers, that's from 4 tons application after 1 year. However, in that same orchard, I continue to apply 4 tons every year ongoing. So every single year I go back to the same plots and apply compost. But for the data you're looking at, that's from application of 1 year.
Okay, thank you. Let's see, and then maybe just quickly, there was a question about the nitrogen release from the compost was 75 to 85%. Was this referring to the total nitrogen in compost or from the organic nitrogen portion?
So the total nitrogen in compost tends to be the organic same. It's almost equal. Because you have to remember during the composting process, unlike raw manure that can have higher concentrations of ammonia and ammonium that then are quickly released, typically compost, when it's aged and cured, the concentrations of inorganic nitrogen are going to be very low. So in the values that I'm showing, it is the total nitrogen pool, but that total nitrogen pool is likely 95% or more in the organic form.
Great. Okay, thank you. Yeah, you have several more questions. If you could answer those during this next talk, I would appreciate it. And thank you so much for your presentation.
All right, thank you.
Okay, so our last speaker for this session is going to be Sarah Light. She is an area agronomist advisor in the Sutter and Yuba County area, and she's going to be focusing on evaluating soil physical properties in response to soil management. Sarah, whenever you're ready.
Okay, well, thank you. I just can't see the timing on here, so can you just let me know if I'm talking for too long?
Absolutely.
Thanks. Okay, thanks so much for inviting me to talk about this today at the ASA meeting. Before I start, I just want to quickly acknowledge that the data I'll be presenting is data from Jeff Mitchell's long-term project, and a lot of it is from Samuel Araya's PhD dissertation, and his PI was Dr. Gazehi at UC Merced. So I don't want to spend too much time talking about the definition of soil health because I already have too many slides, but I do want to focus in on this word to function. Really, what the word to function means is to work or operate in a proper or particular way, to be in working and running order, to perform, to play the role of, or do duty as. We really need our soil to do certain jobs for agriculture and society at large, also known as ecosystem services. These are including but not limited to, and I tried to focus this list on some of the physical attributes of soil, but we need soil to provide space for roots to grow, to provide space for root respiration, in other words, to have enough pore space for adequate air in the soil. We need soil to allow water to infiltrate. We need water to move rapidly through the soil profile when we get a lot of rain. We need the soil to store water for later for crop uptake, and we need it to be able to provide habitat for soil organisms for all the benefits that have been covered earlier in this session. So I want to frame it this way because I think sometimes soil health can be a little bit nebulous, and we're really using these indicators to understand whether our soil has the physical properties needed to do these jobs. Soil is working for us and for society at large. So the data that I'll show is from a really cool 20-year no-till and cover crop project in 5 points. This is, as I mentioned, led by cooperative extension specialist Jeff Mitchell. There are 4 management practices that are used in an annual rotation: tillage and reduced tillage, with and without a cover crop. So there's a standard tillage only with no cover crop, reduced tillage plus a cover crop, reduced tillage only with no cover crop, and then a standard tillage with a cover crop. So with this long-term site, of course, building soil carbon, soil structure, that stuff takes time. And so we're really able to see what the long-term implications of these management practices are. These are in a pretty classic annual rotation with tomato, cotton, sorghum, garbanzo beans, and it's on a clay loam. Soil texture is pretty important for some of these indicators. And so I know somebody asked about that, there was a presentation about the texture triangle yesterday, but soil texture, we're learning a lot about that connection into these soil health indicators. This project has been selected by the Soil Health Institute as part of the North American project to evaluate soil health measurements. For those of you who are not familiar with the Soil Health Institute, I know it was mentioned in an earlier talk, they're doing some really awesome work. They're doing a lot of the legwork of trying to understand which some of these indicators, for example, aggregate stability, which I'll talk about a little bit later, there are multiple ways that indicator can be measured, and some of those methods are very time consuming and or expensive, and some are not. And so they're doing that work of evaluating how the methodology works to measure the indicators and trying to really focus in on what's practical both for a farmer to do, an agronomist to do, what's practical for a lab to do. Some labs are not, a lot of the labs are not doing these really involved soil health methods as the Cornell lab is. So they have 124 sites. 2 of them are at the UC. We're pretty lucky to have 2 at the UC, this one and then 1 at Russell Ranch. And they're really assessing the ability of soil health indicators to detect differences in soil properties. So since reduced tillage and no-till is a little bit novel in California, I just wanted to quickly show these pictures of the practices. We can see that there's a lot more residue in the fields than we normally see in annual systems. The beds are not clean as we're used to, but they've had a lot of success in growing crops for a long time in these systems. And they're direct seeding into residue, tomatoes, cotton, the cover crop here in this picture on the right. So some of the indicators, one of the kind of main physical things that we expect from our soil or that we need from our soil involves moving water and holding water. So I'll talk about available water holding capacity later on. But water movement into the soil is infiltration. That's kind of coming in at the point of entry at the top. And then we also need it to move through the soil. That's called saturated flow. So those are those two kind of important water movement measurements. Some of these can be really just taken from the side of the road. You know, this picture on the left costs $0 to collect, but there's clearly something going on with water being able to move into the soil profile. So we can really learn a lot just by observing after a big heavy rain event when the soil is wet, our water and our soil's capacity to move soil. And I will just say that some of the implications, we may look at this picture and say, who cares? This is a fallow field anyway. But there were this year, we had really, really wet late winter rains, and there were some growers who were next to fields like this who actually lost their wheat crops because there was not enough water moving through the environment in general in the area. And so it might not cause crop loss in your area, but it may cause it in your neighbor's field or in your field next door. So we just want to think about these dynamics generally. In terms of infiltration, there are various ways to measure infiltration. This is called a single ring infiltration measurement method. I'm putting the price here so that you can kind of assess how much it would cost if you wanted to look at these indicators yourself. I think $200 is an overestimate. And I would also just say that time is money and infiltration data takes quite a long time to collect. But there are double ring infiltrometers that are more involved. The single rings seem to work pretty well, so I would just focus on that if you're trying to measure it in your farm. This is just a 6-inch wide piece of PVC pipe that we put a beveled edge on and had a driver that we used with a mallet. Very simple. And then you just have bottles of water. The main thing is to pour a known volume in so that you know what the time means. And then on the right, this is a Saturo device. This is much more expensive, about $4,500. This measures saturated hydraulic conductivity in the field. It's really cool because it can do this complex measurement in situ. You set it up in the morning and then it'll run through the day. So it is expensive. It is a little bit of a pain in the ass, but it's way better than some of the other options. So just to get back to this 5-point site, this data is this is a graph showing infiltration time on the y-axis as compared to management practice on the x. RT is reduced tillage, and then CC is a cover crop. So RTNO is reduced tillage with no cover crop, standard tillage with a cover crop, et cetera. And we see that as we have these management practices combined here on the left, we have the fastest infiltration, which is good. Water's moving very fast into the profile. And then the time takes increasingly longer as we have either just one reduced tillage or cover crop or none of these soil health practices. And reduced tillage reduced the infiltration time really regardless of the cover crops, but the cover crop did have an impact as compared to its comparable tillage practice. And this was taken using a single ring infiltrometer. And the data that I've collected in this regard for another biochar project I used to work on, we also use the single ring infiltrometer. So I would recommend that methodology and not going double ring if you want to keep it easier. Okay, so this is actually looking at the saturated hydraulic conductivity. So again, this is how water moves into a wet soil profile. And this is the same order of treatments, reduced tillage with cover crop and no, and standard tillage with cover crop and no, as compared to the speed. In this case, a higher number is better because it's indicating a flow rate. So it's not actually a time number. And the reduced tillage and cover crops, again, increased saturated water flow, especially in the deeper layers of the soil. Here on the left, we're looking at 0 to 5 centimeters, and on the right, 20 to 25 centimeters. In this case, more water is moving into the soil. Ostensibly, it's going to be available more for crop use later, but really we're trying to get that down in there. And this is also looking at soil water data. This on the x-axis again is the treatments compared to change in water storage. So this data is pretty interesting because it kind of is combining some of the static measurements. So a little bit later, I'll show a graph about available water holding capacity. And those are, that's how we get our moisture release curve, where we know what the volume of water in our soil is at field capacity, the volume of water in our soil is at permanent wilting point, and then that difference, which is the plant available water. So sometimes when we collect our static measurements, we may not actually see differences between the treatments if we're not accounting for conductivity, which is actually water movement in a saturated condition. And so this data set actually, this graph is actually incorporating water retention plus conductivity, and that is what is allowing us to really look at water storage. And again, we're seeing that reduced tillage did have the greatest impact here, with the cover crop was the greatest. Actually, the cover crop with standard tillage, although these are not significantly different, so I can't comment on that, but it is a bit lower. So this is, I think, really interesting because it combines that water storage. And then this is, and just to say that all this data that I'm showing is probably data that would be very challenging, if not impossible, to really collect in a farm scale, like in practice. But at the end, I will talk about indicators that can be used really easily in the farm in an economical and fast way. But this is sort of to just highlight exactly why we care, why we care, and then what are the quick and dirty measurements. So this is looking at pore size distribution. I think this data is particularly exciting. You need to have a pretty involved lab piece of equipment that will visualize all the different pore spaces in the soil and then kind of give you an output. And so this on the x-axis is pore diameter or size, and this is as compared to volume. So it's size over volume, and we're basically trying to understand the area under the curve. So here in purple, we can see that the reduced tillage with cover crops just has wider range. Okay, it has a volume of pores at different diameters here as well as compared to our other management practices. I'll highlight as well these dotted lines. Between these lines are considered storage. Our plant available water that's being held after loss to gravity is held in smaller pore spaces with capillary action. And so those are the smaller pores that are important, as well as these larger macro pores are important for transmission. So that's water movement through the soil to avoid ponding and flooding, et cetera. So a wider pore size distribution is really an indication of healthy soil function. Again, function is the ability of soil to do its job. And so pore size distribution that's wide indicates that the soil can do the job of both retaining water for later and moving water through the soil now. So while we may think and we may use tillage to aerate the soil to open it up, this data actually indicates that the effect of tillage is transitory, and that when we can reduce our tillage and reduce our disturbance in the soil, we're actually going to have increased pore size distribution and porosity in the long term. It's pretty exciting to see it kind of visualized like this. And then just to quickly connect, I know I was asked to only talk about the physical indicators, but I did want to kind of connect those physical indicators to some of the chemical ones. So these three graphs on the y-axis, we're looking at macrofauna abundance. In this first one on the x-axis, we're looking at total soil carbon. There's a positive relationship, which is what we like to see. In this second graph, we see infiltration time on the x-axis as compared to macrofauna abundance. That time that water takes to move into the soil is reduced, which is what we like to see. And then here in this final graph, we're looking at soil stability class rating, which is also called aggregate stability. And that has a positive relationship with macrofauna abundance. And I will just say that there is a plethora of soil chemistry and soil biological data from this long-term research site that I'm sure Jeff would be delighted to share if of interest, but I really wanted to stay to the task of physical indicators. The last data that I'm going to show from this is about dust. This, of course, is topsoil being lost during a wind event, also called erosion. The Dust Bowl was mentioned earlier. Really keeping soil in place is important for the long-term productivity of farms because the topsoil is really our most valuable part of the profile. But also in the valley, our air quality is notoriously bad, and so dust can be a human health concern. And so really keeping as much dust as possible on the field is important, as well as reduction of mites in our neighbors' fields, et cetera. So when they actually measured ducks production by treatment for the standard tillage with no cover crop and the reduced tillage with no cover crop and with a cover crop. And so this is for all the things that were done in the field and the amount of dust coming up. So with reduced tillage, of course, they weren't doing a lot of this land prep, so there's just no dust there to measure. But just to point your attention to these totals here at the bottom, this is cumulative. The standard tillage with no cover crop had the most. The standard tillage with cover crop still had quite a lot, but less. And then the reduced tillage with no cover crop had the least, and with cover crop had more. Of course, there are more management operations with a cover crop than with no cover crop because you have to plant and terminate, et cetera. So that's where that difference comes from. Can I do a time check, May? I can look at my phone.
You've got about 5 minutes, or maybe it's about 7 minutes or so.
Okay, Perfect. I'm just going to fly through these then. Thanks. So when we're looking at this, this is how we used to do it. And then next I'll show something really cool that the Soil Health Institute is doing in terms of developing a model. So these are all pictures I actually put from my phone from when I was a technician at the USDA. So I will speak personally about how tedious and difficult this type of sample collection is. It not only requires quite a lot of attention to detail, a lot of things can go wrong in the field if the samples are collected wrongly, things can go wrong in the lab, and this equipment is very expensive and it takes a long, long time. So that's why what the Soil Health Institute is doing is particularly exciting. But just to kind of show this classic moisture release curve or retention curve. These 4 lines represent different soil textures. Of course, sand has bigger particles, less cation exchange sites, less places for water to hold on to as compared to our clay and our peats, and to some capacity our loams, which have more silt in them. This blue line is field capacity. So that's what's considered held in the soil profile after loss to gravity or drainage. And then this is wilting point, which is kind of water that's being held so tightly by the profile that our plants cannot take it up in the roots. Okay, so plants are not able to extract every bit of available water. They can only extract what is kind of here above this tension. And so what's between wilting point and field capacity is what we normally call plant available water, because we consider that everything in this area is going to be lost to the system before the plant can get it, and everything here is held too tightly for the plant to access. So how we make these curves is collecting these soil cores, and this is an intact soil core which you are collecting basically in the field, exactly representing how it is in the field. So you want to get the structure by volume. Now, how some of the methodologies have been are called core repacking. So people would take soil samples, grind them up, and then repack them in, and then use them on the pressure plates. But that's obviously not as indicative of what's happening in the field because you've removed all the pore space. And so when the Soil Health Institute developed their model, they took intact soil cores and they modeled it compared to some common lab measurements. So they didn't use the grinding core method. And I just want to point that out because when we're really thinking about the details of indicators and methodology, we do have to kind of get into the weeds with this. So anyway, not to move on to the next slide in a second, but you take these cores, you put them on these pressure plates, the chamber pressurizes, and then you reweigh them, and then you know the volume of water that's left in that core, and you get 1 point on your line. Then you resaturate that core, you put it on another pressure plate of a different tension, you wait for the same, you weigh it, then you get another point on your line. So that's how these curves are developed, very tedious. I don't even really know how much this costs. This is kind of old school. Now this high prop machine is over $10,000. It will develop this curve for you with a core. Very, very expensive. I've used it myself. There's a lot of opportunity for error with this. And this is $10,000 for 1 core. It has 1 scale. So not really feasible for us to measure these indicators in our own farms. Now, the Soil Health Institute has developed a new pedo-transfer function to predict available water holding capacity. Soon it will be on the Comet Farm, which is the website that the CDFA uses for the Healthy Soils program, for those of you who have used that before. They developed a model that has soil organic carbon with % silt and % clay. So again, tying that back to texture, and they can actually predict available water holding capacity with an R² of 0.8. This is great. That means we can pull soil samples. It's not going to work in a really freshly tilled field, but we can pull soil samples and we can really understand what our anticipated, water holding capacity is. There are some nuances tying into the chemical and biological. Respiration improves the water, and calcareous soils versus non are seen differently. With aggregate stability, this is a really great indicator of soil health. It's a physical characteristic driven by biology and influenced by chemistry. It really is one of the primary, most important indicators of soil function and soil health. It represents the diversity of architecture. And there are 5 ways to measure it. The Soil Health Institute was able to deduce that the Slates app, which I have on my phone, you can get it for free. And it's very easy to use, very fast. Works reasonably well for me, you, agronomists, farmers, commercial labs, etc. The other 4 ways to measure are very, very tedious. Most commercial labs won't even do those measurements. And so what they're able to show is like, hey, get the free app. It works relatively well. We don't have to do these other very involved methodologies to look at these indicators. To kind of show this aggregate stability data in 2 ways from the 5-point site, this is the aggregate stability measured using the Journat experimental range, which is taking aggregates and dipping them into the soil and then looking and measuring time for kind of dissolution of this, we're seeing that the reduced tillage or no-till, as I put it here, plus cover crop has the highest soil stability class, which means that that aggregate is the most stable as compared to our standard tillage with no cover crop, which is the least. However, when we compare that to this Slake SAP data with the same site, we're seeing the same thing now. These two treatments are slightly different in here, but we're still getting that nice kind of range. It's working reasonably well. Again, I highly will recommend getting this app and using it. You need to use a dry aggregate, air dry soil aggregate. So kind of in summary, the long-term soil physical properties were most improved with reduced tillage and cover crops combined, but individually these practices also did have a positive effect. I know that reduced tillage is very challenging in our cropping systems in California. And so what we can do to build our soil health is very important for anything we can do can have a positive impact, as well as, you know, I'm only focusing on the physical characteristics of soil here, and what the previous two speakers talked about in terms of chemical and biological impacts also can be taken into account when we're selecting our management practices in terms of adding organic matter and soil carbon. So the last kind of measurement I want to talk about quickly, because it's pretty easy to do, is subsurface compaction. In this case, we're emulating our root growth by measuring resistance. And so we want to make sure that we take these measurements when there's moisture in the soil. A root will not grow in bone dry soil, and you will have a terrible time trying to get these measurements, but they're just not indicative at that time. So you're taking them at a time of the year when your root would be growing, at a moisture when your root would be growing. Definitely the easiest and cheapest way to do it was with a shovel. I have diagnosed compacted fields where I've tried to jump on a shovel with my full body weight and not been able to get it in to the profile. To me, that's a sign that there is some serious issues. But we also have penetrometers that can be used. This is one that you can get on Amazon for under $300. The main issue with this one is that this it almost looks like a bicycle pump, but it's like green means go, yellow, and they're red. So you're not getting like a very precise measurement at different depths, versus this one, which is much more expensive and really not necessary if you're just trying to understand the soil function and root growth, but if you're trying to actually collect data. And this will collect down to 18 inches per inch, the resistance, and it will log it for you. So it's much easier. So this is just a range of what we can do in our field. And the last thing I want to talk about before I end is we do have this other new project. We're actually in year 2, funded by the Conservation Innovation Grant Program, USDA, myself, Amber Casey-Ball, Jeff Mitchell, and some people at UC Santa Cruz, Fresno State, Chico State, and a bunch of really awesome and engaged growers are involved in this. Here we are down in Piscinis. These are the sites of our projects, and we have growers who are really, it's really actually very humbling for me to work on this project. It's really been very challenging to try to answer some of these questions around reduced tillage. And there are a lot of risks involved to these new systems that are not much is known, but we have growers who are trying with grazing cover crops, reducing tillage, keeping soil covered as much as possible. These are some pictures from those sites. Here's a picture when we're collecting percent cover of a 6 species cover crop mix that's interplanted into a corn. So the idea is the corn will be harvested and that cover crop species will come up. So really, really innovative stuff. It's a really cool project, and please email me if you want more information about that. I'm excited to hopefully have some results from this soon. So with that, I really want to thank our grower collaborators, as well as Jeff Mitchell, Sam Laraya, Dr. Gazahi, and other people who have collected this data, and my friend Shannon Capelazzi from the Soil Health Institute, who gave me that update on their progress. Thank you.
Great, thank you so much, Sarah. A lot of really great information on research looking at those physical impediments to infiltration and water holding capacity, and there's several questions for you. Someone wanted to know in regard to the cover crop study, I think at 5 points, were they planted as a mixture, do you know? And if so, what was the dominant species?
Oof. So I think that if we look at this, I think it was a triticale rye and pea cover crop is one of the mixes, but what I don't know is if they have used the same cover crop mix every year or if they've changed the composition in time. But I can, if you send me an email, I'm happy to get that information from Jeff Mitchell.
Okay, and then also deep tillage or soil ripping is a common practice, and common practice in Sutter County. Someone noted. or do you know of any research looking at the impacts on some of those soil physical attributes in regard to deep ripping?
I haven't seen data specifically on deep ripping. I know that sometimes we get into a point with our soil where we really need to like get some air in there right away, and that's when we're ripping our fields. I would say the take-home message is as much as we can reduce our disturbance and allow soil function to rebuild, microbes to build that soil structure for us by using the soil carbon sources. In the long term, our soils are going to be more resistant to having deep subsurface compaction. But of course, there are other components into that, like sometimes we have to drive over the field when it's a little bit too wet, and our heavy equipment can cause compaction down at depth too. So please don't take this as in any way of a judgment that we all have to do no-till. I know there are a lot of challenges to that, but when we do have the opportunity, I would suggest that it would be a good idea to try to do it.
Okay, great. Well, it is time to go to break, but you have a few more questions in the Q&A section. So if you might answer some of those, typing in the answers.
Sure.
And we're going to take a 15-minute break before the next session focused on pest management, and we'll be joining back at 11:10. So thank you all.
All right. It looks like we are at 11:10, so I will just get started and kick off the final session of our, of the conference, session 8, Pest Management. My name is Jake Wanger, professor of entomology at Fresno State, and normally this pest management session has been just a smattering of whatever sort of interesting research has come up over the past year or two. And this year, Gina Colfer, my co-chair, and I thought we would go a slightly different direction, basically to sort of dig into this reevaluation of nicotinoid type insecticides that's going on. As you may know, neonicotinoids are currently being reevaluated by the state of California for their relative use in crops relative to pollinator presence. And as such, it's likely that there will be changes in regulations surrounding these pesticides moving forward, and there may be a need in the state for research into alternatives or just knowledge of how alternatives can be used in place of these pesticides. A recent evaluation of sort of the economic impacts came out and found that indeed moving away from neonicotinoids may cause significant changes in the economics of pest management as well, especially around citrus, cotton, berry production, and tomatoes, as well as other flowering vegetables. So this time around, we were hoping to sort of focus in on alternative management practices in these crops, and we managed to find a really excellent row of speakers. I'm very excited to hear from them. We have Mark Bolda discussing alternative controls in berries, Frank Byrne with us talking about alternatives in citrus, and Alejandro Del Pozo discussing vegetable management. And so thank you again to all of our speakers for coming in, for all of you who have stuck around for the entirety of the conference. Much appreciated. This has been a fantastic turnout. And to Gina, who did a lot of the work in organizing the speakers. So thanks to all of you. And with that, we are a little out of schedule, but I don't have 5 full minutes of things to say. So with that, I will pass it off to Mark.
Okay. Can you see me? We good?
Yes, looks good.
Okay, great. Thanks. Thank you, Jacob. Excellent introduction. And also many thanks to Gina and then the staff from UCNR Communication to set this up. So I've been asked to do, I'm going to move these. Hold on. Can I? Okay, let me minimize those. Okay. So I'm going to talk about alternatives to broad spectrum and neonicotinoid insecticides in strawberry. And the way I'm going to do this is in the way of introduction, I'm going to talk about 3 pests. Those will be the Phytophthus mites, then lygus bugs, and vinegar flies. And what I want to do with these is kind of outline one system that has moved away completely from neonicotinoids and broad spectrum pesticides, and another one which is kind of still in a state of flux. And then with the vinegar flies, we're kind of halfway in between. So let's go ahead and get started. I don't have much time to cover these, so I will move rather promptly. Challenges beyond the regulatory. I mean, we're talking about, Jacob, you also referred to this, the regulatory issues with these, but there's others as well. When you're moving away from a broad spectrum chemistry, which most often is very efficacious, to some of these alternatives, we have to make sure that the alternatives work too, right? And then also in the berries, it's a little different than a lot of other crops, is they're harvested quite frequently, between 2 and 3 times a week. And so we can't have long reentries, we can't have long pre-harvest. Really, anything over a day on the pre-harvest interval is going to be a non-starter. And then this is going to be discussed as well with the other presentations, is resistance. We've got to be looking out at resistance in the alternatives as well. How do you move through that? How do you keep a pesticide regime or a pest control regime active and efficacious in the presence of all of this resistance? So let's go through a system which has successfully moved away for the most part from broad spectrum pesticides. And these are the Phytophthus mites, also known as spider mites in our case in strawberries. And one is the 2-spotted spider mite. That's the main one. And then we also have a new entrance. I'm going to spend just a little bit of time talking about here at the end of the spider mite module, is the Lewis mite. I'll tell you how that kind of changes how we deal with this. Alternative methods of control for spider mites are the following. We have predatory mites, looking at miticides, and also the management of the plant and management of the farm. Here's the predatory mite that is very much used. I would say between 90 and 95% of the strawberry ranches use this mite. This is Phytosalus persimilis. It's a obviously orange mite, about the same size as a 2-spotted spider mite, voracious, reproduces faster than the spider mite. And so a lot of people are really happy with it. And so what's happened with this system is that the control and the management scheme has built itself around this mite. So you can't be using broad spectrum chemistries. Neonicotinoid chemicals don't count. They don't work for mites anyway, so they've never been part of the picture. But everything, if it's not compatible with Phytosalus persimilis, it can't be used, okay? So when we go and look at the miticides, I'm gonna just mention another mite here. This is Neosalus californicus. Some discussion in our industry whether or not this one works a little bit better in warmer areas. That may be the case. It's about the same size as persimilis, has a kind of a pinkish cast to it. Again, this is a mite. If its presence is, if it's around, the system will be built around it. So the miticides, moving into the miticides themselves have to be compatible. You'll see I give the name of the miticide, and then compatible or not compatible refers to, is it compatible with these predaceous mites? Agromex is quite an effective material, but it's not compatible, so therefore it's not used very much. Hexathiosox, Savvy, compatible. That's one of the first applications to take place in the season, probably happening right now. Ovicide, bifenazate, acromite, again compatible. Very much a tool for strawberry growers. Esequinocil, canamite, relatively compatible. So you're going to see that being deployed a little bit more to the tail end of the season, where if there's some damage to the persimilis, maybe it doesn't matter because you only got another month or 2 to go. Nealta, again, compatible, so that one's used quite a bit. Etoxazole Zeal, this is interesting. It's compatible if it's coordinated with the Persimilis release. So you can't put it on top of a Persimilis population, but you can, at a set period of time, I think it's something like 10 days, it's now moved into the leaves. Persimilis can be laid on top of it without any ill effects to that population. Oils, not really compatible. There are some exceptions. You see that put as a direct. I found that actually be compatible. And then private industry colleague as well. So the oils aren't in a blanket way not compatible, but you do need to be careful with them. I think that is more or less because it refers to the mechanical control. It suffocates the mites, right? So it'll do both. And so when you look at management of the plant, that also has a big role. And this was work that was done by Doug Walsh and Frank Zalem in the early 90s, late 80s. And a bigger plant is going to shade the plant more. The plant is less warm, and therefore the two-spotted spider mite do not breed as fast, and you have less problems. And this is manageable by the grower because if you don't do strawberries, you're not going to know this, but it's a temperate plant. It does need cold conditioning in order to grow properly. So if that cold conditioning is deficient, then the plant will not be as vigorous, and therefore you have more problems with mites. That was the big finding that Frank and Doug had. Proper irrigation, again, if you're not watering correctly, the plant will tend to be smaller, struggle more, and therefore be more prone to damage from spider mites. So, and good soil management, what I'm referring to here, is more than anything fumigation. Again, if the plant is struggling with pathogens, the mites will find it and you'll have a bigger problem with them. So looking at now management of the farm in strawberry, there's a lot of service roads, there's harvest taking place. There's a lot of things to be done there. There's a lot of roads. And so if you have,
Hi, Mark. Mark, this is Rachel.
Hello. Yeah.
Hi, with PSU.
Yes.
Your internet's cutting out a little bit. It looks like it's, would you be able to maybe turn off your webcam, or maybe if there are other applications open on your desktop?
You bet.
Yeah, okay.
Okay, so the webcam's off.
You sound great.
Well, what the hey? Okay, well, thank you for cutting in, Rachel. I appreciate that. Okay, good. Now I can make funny faces and stuff too, right? Anyway, let's continue. Thank you, Rachel, really. And so dust kills plants. Again, this comes down to the temperature being raised on the plant, and the mites are more pernicious in those cases. Matter of fact, in the wintertime, in the spring, that's where we find our mites are at the edges of the field. They come in where you've got dust on the plants. So that's, in short, that's a system. It looks like I'm locked here. Hold on a second. Just a second, guys. I'm going to have to. Okay. Hold on. Okay. Okay, here we go. So that's a good system that we have for the two-spotted spider mite. It works. It's built up around the predaceous mites, and so broad spectrum chemistries have not been an issue for us in the last 15, 20 years. What to do about Lewis mite? This came in probably 2-3 years ago, I suspect, because we have warmer temperatures. It's not responding to the same miticides, and importantly, the predatory mites are different. So now you've got a new one coming in, and now we need to change the system that we've settled into over the last 15 years. Avermectins, for example, work very well, and how about that? Broad spectrum chemistries work as well. So the challenge for us as research and extensionists is to figure out a solution that doesn't affect so much the two-spotted mites, which are still very much alive and well in these fields. And the other thing too, from an extension standpoint, is how do we get across to people these do look differently, and therefore they need to take different approaches. You'll see on the left, we've got a prime example of two-spotted spider mite there at the top left of the photo. You go a little bit lower in the same picture. Well, those spots are kind of spread out. What mite is that? Because on the right, on the photo on the right, you do have the archetype of a Lewis mite, a little bit smaller, and then the spots spread out across the side. So again, you have a new pest come in, and it causes problems because it can disrupt the pest management system that you've established for the previous. So let's go to lygus bugs. So again, the two-spotted spider mite system is something that we've established. We don't really need to use broad spectrum pesticides, but in the case of lygus bugs, this is a lot more complicated. I think a lot of you are familiar with lygus bugs. Here you have a picture on the left. about maybe an eighth to a quarter of an inch long. And the damage that it causes to strawberries is it feeds on the strawberries in the very early days of its life and then causes this twisting that you see on the fruit. Obviously, that's not a marketable fruit, and it's pretty doubtful that that's even going to be marketable as a processing. Lygus nymphs, lygus bugs do go through incomplete metamorphosis, and you can see they're just smaller versions of the adult. You know, the smaller they are, the easier they are to manage. So we do encourage pest managers and growers to know what these things look like. The one, the one there at the top of the picture looks a lot like an aphid, so we got to make sure we don't mix those up. Here's the problem that we're facing with lygus from a pest management perspective, and it's a continuous problem. It's a California native. It's been here long before anyone was here farming, and it'll be here long after. So you can forget about eradication of laws. Basically, what you're doing as a grower pest manager is managing a crisis. Resistance, so it's been around so long. It has resistance to most insecticides that we use, broad spectrum insecticides included. And then the other problem, I think from the researcher perspective, there's a lack of cohesion amongst the researchers. We have people who disagree on what's working, disagree on what we're supposed to be working on, people actually fighting on stuff. And so it's, this isn't your problem as a grower PCA, but it does slow down the pace of research, unfortunately. So alternatives, this is how we're going to look at the alternatives. So we look at novel chemistries, things that are not broad spectrums or things that are not neonicotinoids. I will discuss here vacuums, parasitoids and predators, and then also trap crops. And some of these are fraught with some controversy, which I'll mention. So novel chemistries, and I think, you can see my pointer here. It is, my take on this is, okay, you're not able to use a broad spectrum as much. Neonicotinoids are, they work, but they work much better if they're mixed with something. Take, for example, in this study here, you have the Octara with danitol. But I think a healthy industry is very well served by an active research program, which is looking at new chemistries all the time. So here's one that we looked at with help from the R4 program. This is Avant and Doxycarb. And then down here, this is one that we got a label for, Sequoia sulfoxaflor. Some people say it's neonicotinoid. It's actually a sulfamide, so it's a little different. Sivanto down there at the bottom of the chart, it does hit the same active site in the pest as neonicotinoids, but it's not neonicotinoid by my understanding. And then there's two there, Belief and Ryman, which actually came out, we got those registered, I think 8-9 years ago. So again, moving these forward. Do the alternative chemistries work? You tell me. You look at the graph there on the left, the bar very much on the left. Right here, that's the untreated control. And you've seen many of these charts. The different letters mean that they're significantly different. So we're getting, on all these alternative chemistries, we're getting significant levels of control. In some cases, really significant. You look at Sequoia all the way over here on the right, it's doing well. And then the thesis that adults are less well controlled by any chemistry is upheld here. You can see that the differences are not quite as wide. In the case of Sivanto, you don't even have a difference. So alternative chemistry. So here's Bugvax. This is a tractor with a Bugvac mounted on the front. You can see this hood goes over the bed. There's a fan in there. It's usually driven by the tractor from the power takeoff from the tractor. And then the bugs are accelerated against these baffles. It's very obvious that the growers is killing lygus. The adults tend to be pulled up easier than the nymphs just because they're a little larger and offer more resistance. But here's the question is, and many growers use them. The Strawberry Commission, for example, has a very active program in looking at these and increasing the power and so forth. But this goes back to the early 90s here. You've got a dream team of entomology researchers here, Carolyn Pickle, Frank Zalem, Doug Walsh, and then my predecessor, Norm Welsh, who basically walks on water. I mean, these are really good people. Tested 3 different vacuums once, then running them once or twice a week. And still at the bottom, and I'll read it for you, the damage in plots treated only with vacuum machines was high enough to be considered economically unacceptable. So they're killing bugs, that's obvious, but the damage is still economically unacceptable. Myself working with our former entomologist here on the Central Coast, Shamat Joseph, we found the same thing. We compared sulfoxaflor with the vacuums, And again, vacuuming method did not provide adequate Lygus Hesperus control and acceptable reduction in feeding damage. Whereas the use of an effective insecticide, in this case afoxiflor, did remain the best control tactic. Two questions. So are we being misled by looking at all of these dead Lygus and so forth, and they're just reproducing at a faster rate? Or are we as academics or scientists failing in some way to pick something up that growers are perceiving? That remains out with the jury. So parasitoids, this one was, it's around. We started releasing this one back in 2003, Parasitenus diganoidus. We're working with Frank Zalem, Charlie Pickett from the USDA. You'll see here one of the places that we released south of Watsonville, a very riparian area. You got the slough right there, lots of refugiae for it. And we did have successful establishment. I remember Charlie, just the excitement that he shared with me in 2003 or 2004, he had found a parasitized pupa. And so they're active, they're around. The problem is, I think a lot of our fields, this is around Castroville, are in this area. I don't see much riparian area at all. And so they have a lot of Lygus, as do the areas that have the parasitoids active. So it's active, it's working, but it's just not exerting enough pressure on our Lygus populations. Likewise with the alfalfa trap cropping, very simple concept is to plant some of the beds at intervals in the strawberry field with alfalfa. Lygus likes alfalfa. They do go towards it. They do reproduce a little bit faster. So you have enormous numbers of Lygus in here. But unfortunately, then we are finding Lygus still in these strawberry beds above the treatment threshold. I think John Longby, another previous entomologist in Monterey County, found threshold, above the threshold, 50 beds away from the alfalfa trap cropping. So, not many people are working with that now. The predator complex, lots of natural enemies in the field. You can see there on the left, Geochris. We got here on the right, the pirate bug, Aureus, right? And the nabits, your assassin bugs, and of course, spiders. And beautiful work done by Diego Prieto. I think he was working at UCSC. Now he's a Lygus guru at Driscoll's, doing very good work there. Found body parts of Lygus in Geochris, in Aureus, and in nabits. They're eating Lygus, but again, they're not exerting enough pressure upon them. The other problem with these is, entomologists are going to know this, these are all related to Lygus. They're all bugs, so they respond to the same pesticide. So if you get good Lygus control, inevitably you're going to be controlling a lot of your predators too. So Lygus, I mean, if it sounds like I'm putting up my hands, I'm not. It's just like, where do we go from here? One of the problems is we don't understand well enough the population dynamics of this pest. We're still wondering where, we know where they come from, but how quickly do they enter the field? In what way do they enter the field? These things are not known. And if you're going to, battle an enemy, you should probably know a little bit more about its habits. The next one is alternative tactics such as pheromones, either to chase them away or attract them to another place. That merits some work. And then also this idea of deployment of vibrational cues. There could be vibrations that they don't like and therefore don't enter the field. That does merit some work. It would be nice to look at that. And then continuing a focus on labeling alternative chemistries. Just continue the evolution of chemistries. Just understand that you're just going to have to keep looking for new chemistries, and you will never settle down, right? You just need to keep running. So the last one, leaving Lygus behind, the last one is this issue of vinegar flies and spotted winged Drosophila, which is also vinegar fly. If you haven't heard of this, I always want to know we're all speaking the same language. Spotted winged Drosophila, the males, it's a vinegar fly. The males have this spot on the back and the females do not. However, the females have a very well-developed ovipositor with which they can penetrate the epidermis of the fruit, which makes them a pest beyond that of just the regular vinegar flies. The problem of vinegar flies, it has a very fast life cycle. Here we have the adult female there in the middle of the picture, and within 10 to 14 days, we've got another adult female. They cycle very quickly, and they have tremendous ability to lay eggs. I think the vinegar fly usually can do 200 to 300 eggs in one lifetime. So they have a lot of ability. So how do we manage them? There's two ways I want to touch on here. One is the cooler, and then the other is in the field. I was a little surprised by this. I was at a Strawberry Commission meeting, I think about a year ago, and some growers did not realize that they're actually killing most of the eggs and the larvae just by putting the strawberries in the cooler. So this was done in wartime Japan, actually, late 1930s, 1940s, comparing infested cherries with spotted-winged Drosophila. Here's a control, so no cooling here. And then you've got your cooling at different temperature regimes, nicely translated. This is from Ed Show. We were working together on this problem back when he was at Driscoll's. So the blue one, 35 to 36, red 34, white 31 to 32. And you can see after 96 hours, i.e. 4 days at all temperatures, all the eggs are dead or no longer viable. Let's just put it that way. Same thing when we're looking at larvae. Again, 96 hours later, 4 days later, all the larvae are gone. So if you don't really have a lot of larvae and don't really have a lot of eggs, you don't really have a problem. The issue is, however, if they get real heavy in the field, you're going to have a lot of damage on the fruit, and you will have some survivors because you're introducing hundreds of thousands of these things into a cooler, and you will have some survivor, especially if the cold chain is broken. So let's look. What is the IPM program that we use to minimize the application of these organophosphates? Broad spectrum chemistries and pyrethroids, again, broad spectrum. Neonicotinoids are not really part of the picture here. So the three sides are detection, use of pesticides and sanitation, detection, trapping. The other thing is, though, having lived, I live here, I work here, I know when these flies are active. It's mid-July every single time. So you know when you should be looking is mid-July, but you can use the traps to get a sense of what's happening in individual field. So control measures, what works? This is all, I'm not making this up. These were all tested when spotted-winged Drosophila came in. We know they work for vinegar flies. They work for spotted-winged Drosophila, same thing. You got to get out there to the field and do the work. Organophosphates, malathion is great. Pyrethroids, Brigade, Danitol, Mustang Max. We used to have Mustang. The factory disappeared in a horrendous explosion in China, so we have to get now the enantiomer, the Mustang Max labeled. Hey, this organophosphate, pyrethroid, broad spectrum chemistries. There are some other things that work in the spinosins and trust organically registered success radiant for conventional pyganic, and that is a natural form of pyrethroid, pyrethrum, probably the original pyrethroid, right? And we also know what doesn't work. Oils, BT formulations, neonicotinoids, and the biologicals, we've really worked with those and haven't found a solution. So sugar enhancement, what happens with sugar enhancement? Flies, when they land on something sweet, they want to feed, but that's not enhancing our sprays at all. And I'll be done here in about 2 minutes. So sanitation is the third leg of this control. So we have detection, we have the use of insecticide. Sanitation is a big one. This used to be a common sight before the entrance of the spotted-winged Drosophila. People just didn't really mind, but now they do, because there's one with a female, and there's another one right there. You can't do that anymore. Sanitation works. This was a work that I did with a local grower in 2011. You've got your blue bars are the ones where we kept it clean, right? Picked up all the fruit, picked the culls out in raspberries, and then the pink is the dirty. And so you can see, even after 2 weeks after starting, the numbers of adults that we're picking up in vacuums there was like a 5-fold difference in those adults. So sanitation works. Is sanitation expensive? Yes, it is, but it does lower your numbers of flies. You can see the malathion applied here in the middle. That lowers all the numbers. And so for a little bit, the sanitation doesn't matter. But then as soon as you get away from that spray, as you see towards the right hand of the graph, there are differences. Program of sanitation, how would you do it? You'd collect the fruit. You can see these are the 2 core containers. And the one right there, right here, is the one that's holding the culls. Take those back and throw them away. Conclusion for spotted-winged Drosophila and vinegar flies: cooling is most of the solution. Be smart and informed about your pesticide use. Broad spectrum chemistries are still in there, but sanitation does work, and do it, especially if you're organic. So some conclusion. We have some systems, as you saw with 2-spotted spider mite, we have some systems that there's no broad spectrum chemistry really needed anymore. However, we're going to be needing to continue active programs in alternative pesticide research just to keep up with the trends in the insects, new ones and old ones alike. active programs and research of mechanical and cultural methods. That's always there, as you saw with the spotted ring Drosophila. You know, the sanitation, some of these questions that we're working on solving with the vacuums, and then recognize strengths and weaknesses of the biological programs. That they're not, you know, it's not, sometimes people think, well, just bring in a fly or some kind of a wasp that's going to take care of it for you, but it's not that easy. There's, a lot of times they're very specific and they're not that strong. So that concludes my talk. I hope I have some time.
Excellent. All right. Well, thank you very
Thank you.
much, Mark. It looks like we have about 3 minutes or 2 minutes. So
Okay,
maybe a
great.
question or two. So looking in the question and answer section, I see down here at the bottom that Rex was asking about, he says that Sean Sweezy's research combined bug vacs with alfalfa trap crops. And I was just wondering if you could comment on the efficacy of those two combined, or if you were familiar.
Yeah, no, I'm familiar with it. Yeah, for sure. So the idea that Sean Sweezy had was to combine the two methods, right? So you know your ligas in theory are gathered on the alfalfa, and then you just need to vacuum the alfalfa because that's where they all are. Well, it's turning out that that's not. You're still above the treatment threshold in the regular field, so you're going to have to vacuum the whole field. That would be my answer to that.
Okay. So you still have to vacuum the whole thing.
Right.
All right. And then we have another question below that. This one says, have you noticed certain insects being more prevalent in higher or lower vegetation areas of the field?
You know what?
And that was.
Go ahead.
I'm sorry, go ahead.
Jacob. Okay. You know what? It's going to depend on the pest. You look at, I didn't discuss this, you know, light brown apple moth. If you're next to, you know, oaks and that kind of thing, you're going to have a lot of them. And that's a real problem because it's a regulatory issue, right? And it'll shut down the field. And it'll cost, you know, it can cost you millions of dollars. On the other hand, you know, if you're talking about whitefly or something, it's irrelevant. So it's going to depend on the insect. We need to discuss the pest itself. And then, of course, this happens all the time. You know, if you have a neighbor who's dirty, for example, if they got a second your strawberry field, they're going to have a ton of ligas. And you know, people have even gone to court on this stuff. So it does depend on what's next to you and who is next to you and so forth.
All
Mark,
right.
One moment. Mark, could you please review
sure.
the answers to the poll questions, the DPR poll questions?
Oh, shoot. Now I just closed it. Can you read it to me real quick?
Oh, yeah. What is the pest management program for 2 spotted spider mite in strawberry built around field? And I am not going to be able to pronounce it, but I do realize that the second
It's
option.
B, Persimilis. Yeah,
Yes.
right. Okay. And what's the next one?
What is the largest way to manage spotted wing Drosophila in strawberry?
So that's cooling. I don't remember the letter. Was it C?
Yep, and most people picked both of those answers. So
All
thank
right.
you.
Okay, good. So even without the video, I was able to deliver, huh?
Wonderful.
All right. And any more questions? Or is that
All
it?
right. Well, with that, we are actually at time. So thank you, Mark, again.
Thank you very much.
And with that, we will move on to Frank Byrne, who will be discussing the use of neonicotinoid alternatives in citrus crops.
Okay, I want to share my screen. Can you hear me, Jacob?
Yes, I can hear you and see your screen.
That means everybody else can hear me then. Okay, good. All right, well, I guess I'm deputizing for Monique Rivera today, so I'm happy to do that. And I'm going to discuss neonicotinoid alternatives in citrus. And I thought I would start by just looking at some of the facts about neonicotinoids that make them so attractive for pest management in citrus. The first one is the mode of action. When the neonicotinoids were introduced in the early 90s, the mode of action was different to conventional pesticides. And with the phasing out of organophosphate insecticides, the arrival of the neonicotinoids were a nice partner with the pyrethroids because they had this different mode of action. So this was very good for resistance management right off the bat. So we can say if we lose the neonicotinoids in citrus pest management, we lose a resistance management tool. So an important question is, are there alternative modes of action? And yes, and thankfully there are some good products coming out now. The second favorable attribute for citrus growers is that neonicotinoids exhibit both foliar and systemic activities. And this, depending on the pest management need, gives the grower a choice to use a foliar or a systemic application. And essentially what you're looking at here is persistence. Now, persistence can be good or bad depending on the pest situation. The systemic formulations, the systemic applications, that property generally promotes longer retention of toxic residues within a plant. And they primarily because they're less affected by new plant growth or leaf expansion, which would impact foliar treatments, and also things like weather. And in relation to systemic activity, they can be active in both the xylem and phloem. And this can enable pest managers to exploit the feeding behavior of target insects on citrus. So, and two of them are listed here. The glassy-wing sharpshooter feeds on the xylem, and the Asian citrus psyllid is a phloem feeder. And being able to exploit their feeding behavior obviously has important connotations because both of these are disease vectors. So looking at some of the current uses of neonicotinoids in California citrus, by far the most important pest that we're dealing with at the moment is the Asian citrus psyllid. It's the vector of Huanglongbing, the causal or citrus greening, which is a deadly disease in citrus trees. There are other pests that are controlled with the neonicotinoids here. I've listed three of them: glassy-wing sharpshooters, not particularly a pest in California, but the glassy-wing sharpshooter overwinters on citrus and builds up to large numbers on those trees. And in situations where you have adjacent vineyards, the movement of sharpshooters into those vineyards can cause a serious problem related to Pierce's disease. And then there's the phytosanitary pest, the Fuller rose beetle and Citricola scale. They're also managed by neonicotinoids. If you look at the kind of use patterns over time, this is the pesticide use report data from the Department of Pesticide Regulation. You can get an idea of, how important the neonicotinoids are within the industry. So here on the left, on the y-axis, we have the acreages, and this is data for Kern and Tulare that I pulled from their database. Imidacloprid on the top shows, it reflects basically early 2000 uses against the glassy-wing sharpshooter. But in more recent years, with the arrival of the Asian citrus psyllid, the use of imidacloprid has certainly increased. And of course, it's also associated with the control of Citricola scale. But certainly the ACP is a major target for imidacloprid. And in relation to thiamethoxam, if you look, it's kind of interesting when it was introduced, when quarantine regulations were introduced to manage the Fuller rose beetle, there was a dramatic increase in the usage of thiamethoxam in order to satisfy the treatment requirements for fruit prior to their shipment to Asian markets. So the loss of neonicotinoids is likely to have perhaps the greatest impact on the Asian citrus psyllid and HLB management. The insect was detected in 2008, and the disease was detected 4 years later. And since its arrival, all segments of the citrus community have been impacted by ACP. And of course, they're all inextricably linked. So the nursery industry, the residential citrus and commercial citrus, all have been impacted by the ACP. And insecticides within each of these sectors of the industry, insecticides are playing a key role in psyllid control. And within the insecticide usage, systemic neonicotinoids, particularly imidacloprid, has been relied upon quite extensively. So why do we need alternatives? Well, the bottom line is because of risks associated with neonicotinoids against pollinators. And the California Department of Pesticide Regulation, the CDPR, it regulates pesticide issues in California. So just remember that, what I just said for the poll. So they have proposed pollinator protection mitigation measures for the use of nitroguanidine substituted neonicotinoids in agricultural crops. And that's basically these 4 neonicotinoids down here: imidacloprid, thimethoxam, clothianidin, and dinotefuran. And the general application restrictions that apply to all agricultural crops is that applications are prohibited during bloom. The application of more than one of the 4 active ingredients, these 4, to a crop within the same growing season is prohibited. And the use of more than one application type, soil or foliar, on a crop within the same growing season is prohibited. So you can already see an impact that the loss of neonicotinoids or changes to their usages could have on citrus pest management. If you apply, for example, imidacloprid for the control of Asian citrus psyllid, that precludes the use of thimethoxam for the control of fuller rose beetle. So what are the options available to growers? Well, we've actually benefited from a lot of early data that has come out of Florida and Texas. They've been living with ACP longer than we have here in California. So we've been able to benefit from their research and their experiences with the insect. And if you look at, this is data from Phil Stansley and his co-workers, dated 2012. And this shows the efficacy of various insecticides listed on the y-axis here, the various insecticides at managing ACP nymphs. And this is by foliar applications. So this is not systemic treatments, this is foliar applications. And basically they measure the relative efficacy of these insecticides compared with the control block, which had no treatments. Now in red here, I've pinpointed the neonicotinoids, and straight away you can see that there are several pesticides, several insecticides that are of equal or better efficacy than the neonicotinoids. And sulfoxaflor was mentioned in the previous presentation. That's a newer compound. These 2 are pyrethroids. Telfenparad is a newer registration. Savanto was mentioned earlier as well. It's a relatively new registration. And so these are as good as, or if not better, against the at managing the nymphs than some of the neonicotinoids as foliar treatments. If you look at the adults, again, you see a lot of products very similar in efficacy to, Actara is probably the most effective of the foliar neonicotinoids. So some of these newer compounds, there's tolfenparad, Savanto again, you've got 2 pyrethroids, but they're highly effective against managing the adult. And often in cases they're more so than the neonicotinoids. So looking at this, it looks like we're in pretty good shape in terms of finding an alternative foliar treatment. Costs will obviously come into consideration, but in terms of efficacy, it looks like there are alternatives out there. So I just want to spend a couple of minutes on research at UC Riverside. Again, Florida and Texas data has helped guide our research. However, California has needed to develop regional treatment plans that suit our local conditions. The conditions in Florida are not exactly the same as they are here. And if you look at the kind of citrus situation in California, this is a graph from, or a figure from Beth Craft and Cardwell, who developed this program for the San Joaquin Valley pest management of pests on navels and mandarins. If you look at the calendar year for citrus, we have basically two major flushes during the year. And these are the major targets for ACP control, because the Asian citrus psyllid must lay its eggs on fresh flush and for effective nymphal development to occur. We need to bear in mind also that there are other pests on citrus. We have the citrus red mite, thrips, scales, and the fuller rose beetle. And during the season, the Asian citrus psyllid is pretty much present all year round. And one of the recommendations by Beth and her co-workers is If you are managing other pests, to try and use compounds that are effective against not just the target pest, but also the agent citrusillid, and then you can perhaps suppress the numbers more effectively that way. So the ACP effective insecticides are shown in red. They're effective against this target pest, but also ACP. And if you move down through this list, you can see as you get later in the season, Admire Pro and Platinum used for Citricola scale, Actara, which is thiamethoxam, Platinum is thiamethoxam as well. It's the systemic version. And then Actara is a foliar formulation of thiamethoxam. But you see the major impact of a neonicotinoid loss would be in this section of the pest management calendar. So in California, the area-wide treatment programs have been developed, and essentially these are the programs that have been developed for Riverside, San Bernardino, and Ventura counties. They're essentially consist of coordinated winter treatments to knock down the overwintering population, and combined with 2 coordinated treatments in the summer and fall. And then sandwiched between those is a grower choice foliar treatment, which would be perhaps targeted at some of the other pests. So again, a lot of this work has been done, and if you consider things, the best management practice that has been recommended by Beth Grafton Cardwell is that the winter treatment is essentially a pyrethroid. There's no neonicotinoids used here. There's 1 treatment. The spring treatment consists of a pyrethroid plus an other foliar treatment, and the timing of these treatments would be at 4 weeks apart. So the first treatment would be a pyrethroid, and there's some examples given here, and the second would be one of these compounds down here. And we saw that some of these on the Stanley data, on the Stanley graph, are actually very effective against ACP. So there are options there for growers to use. Actara is thiamethoxam, so this would obviously be phased out if the neonicotinoids were lost. So, but there are other options. And then for the summer fall, so this part of the management calendar would consist of either one of 2 choices: a pyrethroid plus a systemic neonicotinoid applied together, or the same kind of strategy that was used around the spring applications, a pyrethroid plus an other foliar treatment at a 4-week timing. So, and that would consist of, again, the pyrethroids are pretty much the same as the ones here. The systemic treatments would be Admire or Platinum, and then the foliar treatment would be one of these plus Actara. So, I guess just to kind of cut a long story short is that really a major impact of the loss of the neonicotinoids would be, would hit the systemic section here. So some of the research that we're doing at UC Riverside, and we're looking at these neonicotinoid alternatives using a combination of approaches. So we use lab bioassays where we're evaluating compounds against ACP populations from California. So we need to really make sure that our populations are the same as that are responding in a similar way to the Florida populations. Florida, of course, has resistance, so we need to make sure that our compounds are effective against our local strains. So this is showing the contact activity of insecticides against adult ACP in topical application bioassays. It's a log dose. The log dose down here, and then the response of the insects in the bioassays here. And the black diamonds are basically the response of the insects to imidacloprid. So a very, very steep slope. And we can see straight away that the green and the pink, which are the cyantraniliprole, this one here, and the tolfenpirad, 2 compounds that rated highly on Phil Stanley's graph, very, very toxic to California populations. Then you have Fugimite, which is fenperoxamate, abamectin, which is used widely in citrus in California, and then again Savantov, which is flupyradiferone. all these are very, very effective, but you can see some of the really toxic ones, the cyantraniliprole and the tolfenpirad, looking really good as far as lab bioassay efficacy is concerned. What about the systemics? Well, this is a kind of I call this a colonization bioassay, and in this we compare the ability of the Asian citrus psyllid to establish colonies on citrus seedlings. And this data here is for imidacloprid and thiamethoxam. So these are the two neonicotinoids. And it's essentially a dose response type experiment. So you dose seedlings with different concentrations, and then you monitor the development of the different instars over time. And this is at 2 weeks exposure. We found that we basically define a concentration of imidacloprid or thiamethoxam that's effective against colonization. And comparing two of the newer compounds, cyantraniliprole, which is labeled as Verimark, and Savanto, which is flupyridiferone, we're also able to get a good dose response in this bioassay. The one noticeable thing is, of course, that the concentrations we need that we apply to the soil are higher for the Verimark and the Savanto than they are. But however, we can affect the development or the colonization of the psyllid on those seedlings. If you go back here, note that this is cyantraniliprole. So this is Verimark. This is the foliar version of Verimark. It's called XRL. And so it's showing great toxicity or equal toxicity to the psyllid as the imidacloprid. And the second one, flupyridiferone, which is formulated as Savanto, this is this one here. And you see Savanto is not as toxic to psyllids as Verimark. And you see that here, you need higher concentrations of Savanto to affect the same level of control as Verimark. The other issue, of course, that we'll have to work on is, how do these soil-applied insecticides behave under different soil conditions in California? Because if you have sandy soils or loams or clay soils, these insecticides, the uptake of these insecticides into the plant could be compromised depending on the amount of binding that occurs in the soil. And the goal, of course, is to get effective concentrations into the plant. We've had issues with Admire Pro getting imidacloprid into the plants in heavy clay soils. So this is something we would obviously have to work on with these two new compounds. So just a couple of concluding remarks. We're in good shape with foliar treatments, I feel. There are several new modes of action that show good efficacy in the field. And this is obviously bodes well for resistance management. Replacing the soil systemic treatments could be a little bit more tricky. And we certainly need more research to evaluate the field efficacy of some of the candidates that I mentioned during this presentation. We have a couple of projects that we're just starting that are looking at alternatives for commercial citrus. This project here will do both laboratory and field evaluations of the newer products. And then we have a second project. I mentioned right at the outset that all sectors of the citrus industry are impacted by the psyllid. We've also got funding to look at some alternatives for residential citrus, because they're essentially relying on two compounds: a systemic application of imidacloprid and a foliar treatment of cyfluthrin to manage the outbreak. So if you lose imidacloprid from the backyard citrus treatments, then we could have some troubles. So that's what we're planning to do for the next couple of years. And I don't know how I've actually finished already. I cut down my slides a lot because I was afraid I was going to go over time, but that's what I've got for you, Jacob.
No, excellent. Thank you very much, Frank. That's
It's the first time I finished the talk on time, I think. So, hey.
Well, congratulations.
Yeah.
Yeah, well, it worked out well for us because that leaves about 3 minutes for questions.
Okay.
I saw 2 questions on here. There was one about your figure. It says, in your graph, what do you consider as days of activity for the pesticides listed? I believe that was the graph that was your UC Riverside sort of findings, the one right after Beth Grafton Cardwell's graph.
Oh, this was the data from Phil Stanley, right? This is Florida data. Is that right? This one?
Was that?
Let me see. Let me just share my screen. Can I share my screen again?
Yes, please.
Okay. Let me just get
I
rid of
don't think that'll be an issue.
Okay. I seem to have. Okay. I'm going to share the screen again and let me know if it's this one, then this is not my data. What was the question?
I believe it's the one on slide 13. I think that's where the picture came up. I might be wrong. The question, the person asked a question might be able to correct me. I was a little, let's see here. The question specifically was, in the graph, what do you consider as days of activity for the pesticides?
Okay, I think
so I'm not quite sure.
Yeah, I think it's okay.
This one.
Yeah. The way this graph was, this is the Stanley et al. Data. So the way this is was evaluated was, it was the days when the number of nymphs or adults were lower than the controls. So there could well be, yeah. So there would have been nymphs in some of these, in these blocks. But this is what Phil was doing. It was all related to untreated blocks. So that's my understanding of that.
That's it. Okay, and we have confirmation that was the chart. My mistake.
Okay. Yeah, so it's not
another question we had was.
So I just borrowed it from Florida. So I hope there's no one from Florida going to sue me. So.
Well, they're not complaining in the question.
Okay.
If they are here. Another question was, how do these alternative insecticides affect pollinators?
Well, that's just too long a question to answer here. My recommendation, and some of them are not permitted to be used during bloom. Some of them are. And on each of the pesticide labels now, there is a message about their toxicity to bees. So if the person that asked the question would like further information, then the first port of call is the label of the pesticide.
And I believe the UCIPM has a tool.
Yeah, there's a section there. Yeah, absolutely. I mean, a lot of the newer compounds, the companies are rating their bee safety quite high.
All right.
And
excellent.
one of the reasons for that would be because of the persistence. Imidacloprid, of course, is notoriously persistent in plants, and the timing of treatments is critical so that you don't, deposit the material in the nectar and pollen. So timing in relation to bloom are critical. But those recommendations are all on the UCIPM. Beth Grafton Cardwell has all that outlined there very clearly.
Excellent. All right. Well, we are at time. Thank you very much, Frank. Much appreciated.
All right. Thank you.
Mm-hmm. Frank, this is Rachel with program support. I'm so sorry. Could you identify the correct answers for your 3 poll questions?
Oh, yeah. Question, it's gone again. Question 1 was, which of the following is a neonicotinoid, I believe?
Mm-hmm.
And the answer to that is imidacloprid.
Yep.
The second question was, who regulates pesticide issues in California? That's the California Department of Pesticide Regulation.
Mm-hmm.
I did actually give a hint of that one during because sometimes people say the CDFA, so I just wanted to give a little hint there. And then the third question, there are foliar and systemic options available for applications. So that was the final, the third option there.
All right, thank you so much. And I do apologize for cutting into the next speaker's time. Thank you.
No worries. We went over on questions. With that, we are in the next speaker's time. I'm Alejandro. So why don't we get that screen share up, and Alejandro will be discussing alternative control tactics in vegetables from California's Central Coast.
That's right. Thank you, Jacob, for giving me that pass. All right. Can I get a green light to go? Is that everything okay?
Everything looks great. It just switched.
Okay, perfect. Thank you. My name is Alejandro de Pozo. I used to be an IPM advisor located in Salinas, California. Moved recently to Virginia Tech, and I was able to help understand a little bit about integrated pest management for vegetables in the Central Coast, focusing on vegetables, mainly lettuce and crop crops. So the title of my talk right now, is IPM possible? I hope the answer is yes. And do we have alternative controls? So now it's a little good leeway after the previous presentation. Can we think about what are the alternatives besides insecticides that we can use? And I'm hoping that I can convince you that we have some alternatives, at least in the vegetable production, use vegetable production as a study case to show those alternatives. So that's a beautiful shot of romaine lettuce field in Salinas, California, in the back, the hills. There's a harvesting crew working on the way. So as all the agroecosystems, there will be populations with insects that are going to be problematic in our perspective as growers, as BCAs, or consultants, and we need to manage those populations to not getting into damage the crop, either the cosmetic damage or the actual injury caused by this insect. So, and when I talk about that, for example, this is a great shot of the damage caused by aphids on romaine lettuce. So on the right hand side, you have a healthy lettuce compared to the one that is heavily infested with aphids. And yeah, I don't think any of your consumers, like the end users or consumers, were going to get the one from the left. So we, as IPMers, we would like to have our programs lowering those populations and keep everything on track. Not only aphids are a problem in lettuce, and not only lettuce, but also crop crops in Salinas, California. You have also thrips. And one of the things that thrips are doing in the recent year, couple of years, is vectoring the INSV, the impatient necrotic spot virus. And that's something that actually can have 100% damage rates when you have high infestations of thrips vectoring this INSV virus. And then for cold crops, you have the diamond packmo, this caterpillar that is a nuisance. It can create like the indirect effect when it defoliates, and it creates all those holes on the left-hand side. But also, when you don't control these populations, the caterpillars will jump into the heads of the actual finer product, talking about the head of the broccoli or the cauliflower. And then on the bottom right, you see those nasty holes and chewed up and frass that no one is going to let it buy if you're harvesting top premium product that needs to go to the shelves. I like to take this opportunity to talk about IPM as a concept. I mean, everyone on this webinar will be at least once been asked or think about what is IPM. So I want to share with you what the definition that I've been using through the past of the years, and I really like this one. So it's the one that the National Coalition of IPM in '94 come up with. So saying that IPM is sustainable, and I highlighted well, actually bolded and underlined that because we want to make it for, true time. And it's approach the managing pests by combining, and that's the key. So you're using multiple tactics. Sure, sometimes insecticide use is the first one to go, but can we consider something else? And that's kind of like our responsibility as IPAers. And we're combining biological, cultural, physical, and chemical tools. And this is where the part that I really like with this definition, because it helps, IPM helps to minimize economic. So to be frank, this is something personal. We're all doing agriculture for a business, and something when they start touching our pockets, it's not something that we would like to see. So this definition encompasses that. Don't get me wrong. Don't quote me on this. Alejandro Esposa, that I'm not trying to say that I don't care about the environment or something else. But as being part of the agricultural community, we need to be aware that there is economic, cost associated with all of the decisions. So economic health and environmental risk are prone to be, minimized using IPM. Professor Pettigo back in 92, in the 90s, envisioned IPM as this beautiful bridge that you can build to not being drowned in the river pest losses. So if my mouse is correct, you have your keystones. That's the information that we need to know. That's why you guys are here in this webinars and going to, the extension seminars and anything that's organized to learn more about these insects that are actually problematic in our systems. And then on top of that, you build these pillars. Those are the tactics that you're going to use to create this beautiful bridge that it's called the integrated pest management. Do we have an IPM in vegetables? Yes, the answer is yes. Salinas growers have been doing this for several years. And the idea is that there's three main pillars: cultural control, biological control, and chemical control. So within cultural control, you have all these practices that you can do: selection of varieties, doing the tillage of the ground, selecting the areas, avoiding hotspots from last year, for example, in thrips management. Those are kind of the cultural practices. And then we're going to talk about a little bit about conservation and biological control. And then we ended up talking about some of the chemical control. Well, one of the, the first steps is we need to know what's out there. So we need to actually walk the field. I really take my hat off for all you guys, if you're PCAs or people, you're the growers, are walking every day those fields. And it grinds my gears when, other community members will say, well, the growers and the PCAs, they just spray pesticides because they want it. They like to spray. They just keep spraying. That's not the truth. You guys are so highly professional that your professionalism is so high that you guys actually walk those fields and know what's going on and decide what to do and to reduce those populations of the problematic insects. Well, something that it can help you manage your time, because now you have more acres and more acres to walk. Sometimes it gets a little bit impossible to do that with it. It becomes a single man job. Well, you have all this additional tools that will help you. One of the examples is this smart traps. They are high resolution cameras put on the trap. You use a pheromone lure to attract the adults. And basically that has a cellular modem that connects it to a computer that actually based here in the East Coast. This is actually something that I really liked it because it helps the growers to understand what in real time. One of the biggest drawbacks of using pheromone traps is like, well, Alejandro, we're just gonna get the data from 7 days ago. Well, not this trap though, because this trap will send you alerts every day. And the idea is that you are kind of like in a real time. Yeah, I don't wanna learn more about, I don't wanna be stressed out. Well, that will be the problem. Now there are massive amounts of data there's gonna be thrown at you, and now how you can manage those things to make more informed decisions. So one idea is to do pest remote monitoring, that you got all these different traps throughout your farms. You can understand when the peak of activity of these insects are around. One example is the diamondback moth that we have inactive actually right now as we speak in Salinas, California. What it does is this computer with those images will recognize the moth that will count it for you and then send you an alert. So here in the picture is actually what the computer is looking at. And those green squares are the ones that the computer recognizes. Those are the new. And the lighter green, like the thinner squares, are the ones that were recognized from the night before last night. So that's how the computer keeps track of, how many are getting new captures and then send you those alerts. Now within the second pillar, you got biological control. It's good to be aware that there are good guys naturally occurring in our systems, and especially in lettuce, we were able to see ladybugs, parasitic wasps, lacewings, but the king and queen of the doing the predation services is the hoverflies. Hoverflies on the bottom, you have the adult that resembles to this bee-like. But actually, it's a true fly. So how tiny those eggs are, and then the maggots are actually the ones that are going to predate on the aphids. Some here and there, sometimes we're going to see, at least in Salinas, California, sometimes we'll pop this entomopathogenic fungi that's going to infect aphids and create some episodic episode. When I have a time over there in Salinas, I was able to understand this a little bit better. So Salinas, California, the growers there are a world-class example of using conservation biocontrol, incorporating insectary plants to attract beneficials. So the idea is that you create shelters and you provide alternative food to the beneficials that are flying around to attract them and therefore stick around in your field. So they have both different approaches. You can do strips throughout the field, or you can also scatter the plants and when you do transplants and things like that. So I was able to team up with a group of growers and PCAs in Salinas back in my time, and we were able to understand what is the influence when you have these insectary plants. So we selected 12 different fields, and we were able to follow up those fields and see the population fluctuations of aphids in terms of if you have the insectary plants or if you don't. So we have 3 different treatments back then, where we have fields, organic fields with no insectary plants at all, and then one that have the alicin, and other fields that have alicin plus something else planted in the field. So our results indicated overall across 2 years in 12 different fields that we were able to document fewer aphids in those fields that have the alicin flowers. You see here in the graph, you have the gray bars that actually have the aphid population, and then you have the black bars that have the maggot, counts per lettuce head. We saw that the ratio, so the number of maggots compared to the aphids was significantly higher when you have this alicin plants being incorporated in those lettuce fields. The control plots were higher compared to those alicin, and what was surprising is that when you start incorporating other plants, at least the ones that we looked at it, you can probably lose a little bit of that only alicin effect for attracting hoverflies. I'm not saying here that combining species are not a good practice. Back then, that combination that we used was already pre-made, probably didn't benefit that. So there might be more research to do to find if there is any potential combination that can promote the presence of this guy. So combining insectary plants is not a bad idea, but at least the ones that we use here, or the ones that were used, didn't promote that presence of them. So overall, at the end, we were able to see some consistency presence of these maggots here in the fields that had the insectary plants compared to the ones that did not. Another option when you are talking about biological control is releasing beneficials. And in the past, and I've done it when I used to be a grower back in Peru, my home country, is we have this crew, this army of people shaking the containers and dropping beneficials through the field. It's so expensive, it's not consistent. Now with new technologies using drones, you can use the drone to release the beneficials. So we team up with a local company trying to understand if that is an accessible and actually reliable way to do it. It turns out you're going to see it. So what we can do is you can target field perimeters. You can target also the interiors. Not necessarily you can do blanket applications, but you can also with the drone program the pattern of the flight and actually target specific areas with a heavy release, since you know there's a hot spot. You can release also beneficial in the insectary plants, or if you have trap crops, you can release beneficial in those trap crops. So the species that I used to work with back then was Aureus, lacewings, and the amblyopsis predatory mite. More work I was able to done with lacewings and the predatory mites specifically. So here what we did is we set up some collection stations, trying to understand how, if we're losing some of the the material that is coming drop from the sky and dropping by the drone. And it turned out that yes, there is a percentage that would be placed on the ground, but more of like at least 70% of all the content it's being dropped on the canopy of the plant canopy. Remember that so when we're releasing these beneficials, is the canopy, at least lettuce or the other vegetable crops, the canopy is really kind of like getting close. So you're actually not dropping too much of that content on the ground. But for our surprise, when we analyzed the content from these containers, these stations, we were able to find, so you mix lacewing eggs with the rice hulls as an inner material, like the media to drop those lacewing eggs. And I was telling my team back then, is like, there is no way they want to find anything. It's just the rice hulls. Well, to surprise, when we go to the lab, we were able to find viable eggs on several of these stations that we put it out, and those eggs actually hatched to lacewing. So it was a great surprise that we didn't expect to see. And actually that's what explained that some of the growers are actually releasing beneficials using this technology. So we team up with that grower in Salinas and Soledad, and we were able to see that when you release beneficials, you can reduce populations of aphids significantly. Is it enough? No, it wasn't enough, because when we finished these trials, the grower needed to come back and spray those experimental plots, because still, when you're looking at 100 aphids a lettuce head, that it's impossible. That is not, that's not acceptable. So is this a silver bullet? No, it's not. That's why it's called integrated pest management, because you can release the beneficials, lowering your population. So we did it in 4 different fields. We see some consistency when you have higher aphid populations, so it might be a functional response about that. But it was neat to see that if you release beneficials or you spray certified organic compounds, you should be able to reduce populations of aphids, at least under commercial conditions. We also release predatory mites in ice plant patches. So ice plants are a roadside, like more like a preventive erosion in some of the areas in Salinas. And the idea is that you have those on the perimeters of the farms, and they always have blooms and flowers, so that attracts the western flower thrips. So we see those patches of ice plant as trap crops. If you do nothing about it, the thrips will gonna, infest those plants and then reproduce, and they're gonna like become like a nursery. So we decided to test those and release with the drone predatory mice to lower those populations. So we were able to see that compared to the control ice plants that we don't release anything, there was some reductions, not extremely significant in terms of the numbers, but we were able to see the populations were reduced. It wasn't the only treatment that we needed to do. That is correct. So we needed to explore that. This trial was exploratory. Something that also we learned about it, and we were able to actually recover from the ice plant flowers the predatory mites when we collected those samples. That was really eye-opening. That means that you're releasing, you're gonna be having those predatory mites in the actual ice plant patch. And lastly, one of the techniques that it could be thought incorporated in your IPM program is mating disruption. If you're talking about cold crops, we did this. The idea is that you release these pheromones to attract the males in order to confuse them. So the attraction is for the monitoring trap. In this case, you inundate the landscape in order to confuse the males. The idea how to measure success of mating disruption, one of the proxies will be that you have a monitoring trap in the middle of the experimental plot, and you will see 0 captures. That means that the males are flying around, they're not able to find that specific pheromone trap. That means they're really confused. So when we did this, we were able to show that this have some prominent results, at least promising results. When we did it, and we put 32 of those, did you see those stakes with the dispensers, the solid dispensers, they have a release rate of the pheromone. We were able to see when you put 32 of those dispensers per acre, we were able to shut down that monitoring trap compared to when we put 16 or when we put nothing. That will be the controls. And then when we analyze some of the damage of the caterpillars, there was a reduction that we observed after a specific, 7 days after our second evaluation, when we put all the dispensers and we were able to see that the number of holes or damaged leaves were reduced throughout the effect, especially in the plots they had, the 32. So one of the goals was to understand if 32 dispensers, it's a good number. It seems that it needed to do more replications of that, but unfortunately, COVID hit and we were not able to complete that study. But actually, this preliminary pilot showed us that it could be something promising to incorporate that, since diamondback moth is something that worries the vegetable growers. And with that, again, why should we care about IPM? Yeah, we have the problematic and costly to manage this pest, lettuce aphid, western flower thrips, diamondback moth. Why they're problematic? Because there are huge populations of them in our crops. They persisted through the seasons. They're always there. And on top of that, we're going to have regulations on insecticide use. IPM is possible. Hopefully, I will show you an example that it is possible to use different tactics besides chemical applications that can be, incorporated in your program. And that includes cultural practices, biological control, and chemical tactics. And with that, I'm going to finish with a nice shot of Jasmine throwing some bags for collecting samples to Richard Smith when we're doing some thrips and INSV sampling monitoring back then in the old days. So with that, that's it.
Excellent. Well, thank you very much, Alejandro. So despite starting 3 minutes late, you actually wrapped up more or less right on time. So very nice.
Yeah.
We have a handful of questions in here. The first two both relate to the intercropping you discussed with the sweet alyssum.
Mm-hmm.
And the first question was, one of the was what were some of the other insectary plants that were grown with the alyssum? And then a follow-up with that was basically whether the presence of these special insects, particularly the fly maggots, reduced the quality of the vegetable crop for the buyer.
So for the first question, for example, one of the growers, he used to use Cosmo, and it seems like Cosmo is not a plant, in my opinion, that might be considered to be incorporated in. But if you're talking about other alternatives, cilantro, dill, this white flower plants could be an alternative. We didn't have the time to look at those alternatives. So a research question that was pending is, are there any other alternatives that can be incorporated as for the sweet alyssum? And then for the second question, yes, sometimes it could be problematic when you have high populations of aphids that you're going to end up having maggots. I mean, long story short, when I was in Salinas, people will complain having ladybugs on their salad packages. How can you tell an end consumer that having a ladybug inside the salad is not bad? It's actually kind of like the opposite, but bug is a bug. So sure, it could be problematic to have beneficials in your field.
Of course. And with that, we are at time. There are 2 more questions. If you would like, Alejandro, you could take a look at those and answer those in the Q&A box. But beyond that, we are at the end. I see that the poll results are being shared. So can people see that? Or do we need to go over the answers again vocally?
If you could just identify, Alejandro, the correct answers for questions 1 and 2.
Yeah, the first one will be all of the above, and the second will be true.
Lovely, thank you.
Excellent. Well, so with that, we are again at the end of the conference. So not again, but we actually are here for the first time and the last time. So with that, thank you all for coming and for sticking around as long as you did. I think this is the biggest attendance I've ever seen at the pest management sessions. So I'm really glad to have you. Just a reminder before you go to please fill out the evaluation forms if you have not already done that. The evaluation forms are really important for us in terms of what topics we're going to choose for next year's conference. It helps us with selecting our honorees as well as identifying potential new board members. So your input is evaluated. We really read these things and we rank all the results and to come up with next year's conference. So if there's something you really want to see or someone you think really deserves if there's anything that really needs to be brought up or someone you think deserves to be honored, please let us know. I have a note here that when you fill out the evaluations, they can be emailed to Rachel. Hopefully I got that clarification correct.
We actually
right.
have, when you close out your Zoom webinar, the evaluation link will pop up hopefully in a new browser, and you'll be able to fill that out online. And if you don't end up receiving it, if say Zoom doesn't open in a new window for you, we will send it out in an email no later than 5 p.m. tomorrow with the CCA, DPR, and CURES instructions.
Excellent. So it sounds like you'll have access to it as soon as you close out. And similarly, it will be emailed to you. So please, again, just take the time to fill that out. It's much appreciated. All right. Well, with that, thank you again for coming. And I'm just going to step away.